All right, good afternoon and welcome. I'm Marianne Hamilton Lopez, Senior Research Director at the Duke Margolis Center of Health Policy. Welcome to today's workshop, Advancing the Development of Therapeutics through Rare Disease Patient Community Engagement, which is being convened by Duke Margolis and our partners at the FDA. As many of you are well aware, 7,000 to 10,000 rare diseases collectively affect approximately 30 million people in the United States. These diseases vary widely, but many are progressive, serious, and life-threatening, and only a small percentage of these conditions have approved treatments. Developing treatments for rare diseases is challenging. Small patient populations can make it difficult to power clinical trials. The natural history of disease may not be well characterized, and symptoms and disease progression may vary significantly within a given patient population. Given these and other challenges around the conduct of clinical trials in the rare disease space, patient and caregiver engagement can play a vital role in supporting therapeutic development by helping to fill knowledge gaps and ensuring that the clinical trial and review process is informed by what is feasible and meaningful for those living with rare diseases. In recent years, we've seen encouraging progress in engaging patients and caregivers early and often throughout the drug development process, thanks in large part to the tireless efforts of patients and caregivers themselves. There is an opportunity to learn from existing strategies that have been effective for incorporating patient and caregiver input and identify where future and further improvements can be made to best support rare disease drug development and review. It is important to ensure that patient engagement activities are informative and productive for stakeholders engaged throughout the drug development and review process, and that such activities are not overly burdensome to patients and caregivers. And it is critical that researchers, regulators, and industries communicate out often what patient experience data is most helpful and how such data shapes thinking and decision-making in a way that results in timely development of safe and effective therapies for rare disease patients. That's why we're all here today, to discuss how to best understand patient experience living with a rare disease and how to incorporate those experiences and priorities throughout the drug development process. This includes understanding patient perspectives on the burden of their condition and any existing treatment options, as well as how their current health status and risk of disease progression may impact willingness to accept risk from treatment side effects. Next slide. And with that, I want to take a minute to review the sessions we have lined up today to explore these topics. First up today, I'll be joined by Dr. Peter Stein and Dr. Nicole Verdun from the FDA for a fireside chat on how FDA has been working to incorporate input from patients and caregivers in their work, and importantly, how ideas and input from forums like this one can inform FDA's work moving forward. We'll then hear a brief set of presentations from FDA on the drug review process and how early engagement with key stakeholders can provide important information to inform FDA's regulatory advice and decision-making. With that context in mind, we'll move on to a session focused on trial design and related aspects of drug development. As I mentioned earlier, clinical trials for rare disease treatment can pose unique challenges, and meaningful patient input in trial design and execution is critical to ensuring the process works well. We look forward to panelists' insights on these topics. Next, after a quick break, we'll have 3 case study presentations highlighting how patient community engagement has helped to answer research questions at different stages across drug development, followed by a panel discussion to explore different approaches and identify opportunities for improvement. Finally, we'll wrap up with a discussion we've titled Where We're At and Where We're Going, in which panelists will share reflections on how approaches to patient engagement have evolved and provide recommendations about how such efforts can continue to improve in the future. Next, before we get started, I'll review our statement of independence. The Duke Margolis Center for Health Policy is part of Duke University, and as such, we honor the tradition of academic independence on behalf of its faculty and scholars. Neither Duke nor the Margolis Center take partisan positions, but individual members are free to speak their minds and express their opinions on important issues. Also, please note that today's meeting is not a Federal Advisory Committee meeting. While we are keenly interested in your input on the topic, we won't be following advisory committee procedures or taking a vote at the end of the meeting. The meeting will be a success if there's an exchange of lots of ideas and open discussion. Finally, I'll remind all our speakers and panelists to include any relevant disclosures or potential conflicts of interest in their remarks. Now, we have just one more slide with some virtual meeting reminders before we get to the opening fireside chat. All attendees are encouraged to contribute to the meeting through the Zoom Q&A. We have set aside in each of our panel discussions to bring in audience questions. So again, we welcome your submissions via this function, and we'll try to answer as many as possible with the time we have. We'll also be using some audience questions that were submitted in advance via the web form on our registration page. So thank you to registrants who sent us questions there as well. I'll also note that FDA has a docket open for further comments regarding this meeting and the topics covered here. The link to that docket can be found on the event page on the Duke Margolis website. And we encourage attendees to share feedback via public comment. We are recording this meeting, and we will be posting the recording on the Duke Margolis website for future reference. The slide deck will be posted following the meeting as well. Now, it is my pleasure to introduce Dr. Peter Stein, the Director of the Office of New Drugs at FDA's Center for Drug Evaluation and Research, CDER, and Dr. Nicole Verdon, Director of Blood Research and Review at FDA's Center for Biologics Evaluation and Research, CBER, for our opening fireside chat. I asked them both to come on screen. Wonderful. Well, thank you both so much for joining us. For our first question, I'd like to ask if you could both elaborate on the valuable role of patient input in forming the drug development and review process. More specifically, how is information shared by patients and caregivers, and how is that balanced alongside the evidence throughout the drug development and review process?
Sure, I'm happy to start. Well, first, I just want to say I think this is an exciting and important meeting, as we'll be discussing the role of input from patients, which is critical throughout the drug development process and is essential in FDA's regulation of drugs being developed. So hearing ways we can enhance that input will be highly important and impactful. FDA is committed to doing all we can to further the development of drugs for rare diseases. These are devastating diseases with profound impacts on patients and families, and unfortunately, as we just heard, Marion, as you emphasized, most lack approved therapies. It's important to emphasize that we all share a common goal, which is to bring effective and safe treatments to patients as quickly and efficiently as possible. So in that regard, patient input is essential throughout drug development. And you'll hear discussions of this throughout the meeting, but I'll touch on a few key points. Input from patients is essential even before the first step in the development of drugs, and that's in understanding the disease itself and how it affects patients. The burdens that patients experience, the benefits and problems with current treatments, what they view as the most troublesome symptoms. Collecting this is vital information as a key objective of the patient-focused drug development meetings, as well as other listening sessions with FDA, with patients and families and caregivers. Mm-hmm. I'd add that many clinical staff in O&D and in other parts of FDA remain active in clinical practice, and so caring for patients and hearing directly from them about their challenges and experiences with their disease. Patient participation and patient stakeholder group engagement and collaboration with registries and natural history studies of a disease is an essential way to help in our understanding of the disease course, its complications. And this information is absolutely critical in the design of the controlled clinical trials of drugs. And these controlled clinical trials are an essential tool that we use to evaluate drug candidates. And patient input is critical in the design of these clinical trials, both in the overall trial design and particularly in the crafting of trial endpoints. We put out 4 patient-focused drug development guidances to advise drug developers on how to obtain critical information from patients on aspects of disease that most impact how patients feel or how they are able to function at home or school or work, and how drug developers can translate this information from patients into endpoints that most accurately measure these aspects in the setting of a controlled clinical trial. The patient input on trial protocols is also important in ensuring that the design is feasible, that patients can participate and remain in the trial. And developers routinely meet with patients and their families to get that input and review these protocols. And of course, patient stakeholder groups are often important partners in helping to recruit controlled clinical trials. Now, results from the controlled clinical trials are the critical information that FDA relies on to evaluate the effectiveness of the drug. And does it have the benefit that it's anticipated to have or proposed to have, and also to assess the drug safety profile. And for all drugs, we have to conclude that the benefit the drug offers patients outweighs its risks and that there is a favorable benefit-risk balance. All drugs must have substantial evidence that they're effective to be approved and a favorable benefit-risk balance, but we recognize that for drugs for rare diseases, we must accept more uncertainty than we would for a drug for a common disease, for example. And understanding from patient input as to how patients value the benefits the drug offers and what risks from the drug they can accept is essential in our thinking about this assessment of benefit and risk, and also in our acceptance of some uncertainty, even as we still must conclude for all drugs to be approved that there's substantial evidence from controlled clinical trials that the drug is effective.
Mhm.
So the role of patient input starts before drug development and continues to be essential throughout the development through FDA's review of the drug and our decisions about drug approval. So really from the start, or even before the start, right through the work that we do to review the data from a drug, and even after the approval of drug, input from patients is absolutely essential in the process.
Excellent. Thank you for those opening comments. Nicole, I'll turn to you next.
So thank you. I think that was very comprehensive in terms of his comments. And so I first would like to just thank you for having me here. When I saw Duke, I was quite excited because I actually went to Duke for undergrad. So that makes it even more special. So I would just add, in addition at CBER, we are really very quite focused on the patient and the patient voice. And we do that in several concrete ways. One is that we really do encourage sponsors and investigators to collaborate with patients directly when they're designing their development program, and get their input in really identifying what's meaningful to the patient, what's meaningful to the caregiver. And so we bring that up quite early in the development process. In addition, in gene therapies, I think it's uniquely important to make sure that we're getting patient perspectives. And we actually hold meetings to sort of get to that with including public listening meetings. We had one in particular, patient perspectives on gene therapy products, that was quite helpful. And we take this advice quite seriously and try and incorporate that into the decision making that we have. We also have town hall meetings in this space as well around some of the rare diseases and including patient engagement. And so I think that just to underscore, without adding too much, I think that the patient voice is integral into the development process, and we do it in several ways, both proactively as FDA and in the Center for Biologics, and in addition, actually trying to hold the sponsor to that same engagement. So I appreciate that question as a start. Thank you.
Thank you. Nicole and Peter, you both gave some examples of both current and developing efforts at the FDA. Are there any that you feel most resonate or are the most promising for increasing meaningful and impactful engagement with patients? Based on the various examples, is it a one-size-fits-all, or are there some that really differ based on the various communities?
That's an important question because it's not going to be one size fits all. I mean, clearly, the patient-focused drug development listening, these sessions have been very, very impactful. I know many FDA staff, particularly in the divisions that are relevant to that particular disease, but across our organization, attend those sessions and are very much affected by and learn from those sessions what is meaningful to patients, what are the ways the disease is experienced impacts their lives, impacts their families, that's most important to them. We've had some really important learnings that have changed how we've thought about what the most important targets for disease or what symptoms are relevant as we try to review the data and as we give advice to companies in the design of the trial. So those sessions are incredibly impactful. But other patient listening sessions, smaller sessions that we get to meet with patients and families are also very impactful. And I'd also say that input and advisory committees during the open public forum, open public hearing are also very, very impactful to us. We hear that, we see that the advisory committee members pay attention to that. And I think that really is input to us that does influence our thinking about how patients are viewing the benefit of that particular drug and the risks as well. And we just announced the formation of the new advisory committee for the Genetic and Metabolic Disease Advisory Committee, GEMDAC, which will be a really useful setting for discussions about drugs we're reviewing for rare genetic and metabolic diseases. But a place also, I think, during the open public hearing to hear from patients about their experiences with those particular diseases, and of course, to hear from the patient perspective from their representative member on the committee. So I think there's a wide range of different settings and forums in which patient input is important. And some of those are formal as well as informal. We get emails, we get calls from patients who want to share their experiences or tell us what their thoughts are about the general development in the area of disease that their family is experiencing. And that input is always very helpful to us. It influences our thinking and as an important context for the rigorous review of data that we have to pursue.
So to answer the question about specific things that might be more helpful, I think all of the engagement, as Dr. Stein says, is helpful. But I think in particular, when thinking through that question, what is particularly helpful is with individual diseases in the rare disease space, having conversations within the disease community about endpoints and what's important, and coming to consensus within that as much as we can about what that looks like. And so the reason why I highlight that is in the rare disease space and sort of thinking through some of the development programs, what helps is when the community is on the same page about what they think are meaningful endpoints. And so to speak to something specifically, I would highlight that. I do agree that all of the engagement is meaningful, but particularly in the development space, when we're trying to get things to market as soon as we can, I think that's important. And, with a lot of the rare diseases, there's significant heterogeneity within a population that can sometimes make development a bit challenging. And so having some consensus and conversation around endpoints is, I think, quite helpful.
Both responses are incredibly helpful to us. As you know, this meeting will focus on how to best understand a patient's experience living with a rare disease and how to incorporate those experiences and priorities throughout the drug development process, which I can imagine would be quite an undertaking for some of these communities to figure out how to engage and where to put their efforts. So, one of the questions Peter, you said that this will help influence our thinking. And Nicole, you're talking about, how this would really help the agency think through endpoints that could be put into practice. A lot of times when we think about research or asking people to engage in research, they wonder, how will their voices be heard and how will it be used? So I guess my question here is, how may these recommendations and the feedback shared by patient communities and other stakeholders inform like this public workshop or through the public comment docket? How does that specifically inform FDA's work to support rare disease drug development and the review process forward? What information can we give back to those communities about how their efforts are really working to engage your thinking as you're making these really difficult decisions moving forward?
Nicole, I think I've been jumping in first, so I'm going to let you jump in first, and then I'll jump in afterwards. So please.
So I think a specific example I can highlight are the approvals on Friday for the first gene therapies for sickle cell disease. We held a patient-focused drug development meeting in that space a few years ago now. And we really listened to the input of the patient, what was meaningful to the patient, what endpoints were meaningful, and then specific conversations around pain and to use an example, and how to measure that pain. And we incorporate that into the feedback that we give to individual sponsors in terms of endpoints and otherwise. And I think it's a huge a wonderful milestone to have these two gene therapies approved in a disease area that has had limited progress overall in treatment paradigms. And so I do credit the patient community and patient engagement in that space. And so I just encourage, sometimes I think that it's not always transparent about where that voice is, right? And how we listen to that voice. But I want to underscore the importance of the voice and the fact that we do definitely listen to the patient voice. And specifically, I think that the agency, we are discussing in CBER ways to be more transparent about the use of that voice so that people can understand that in real time, because we don't want to lose that voice, and it's quite important.
Yeah, I mean, and I would just add, because I think really touched on the key points already, but what I'd add is that as I said, this is not a one time, one point in time input. It's really an input that for the diseases that are these very challenging diseases that we hear about the patient experience throughout development, because there are so many different parts of this. As I said, understanding the disease in the first place before drug development even gets going, making sure that the patient voice is captured in the design of the trials is absolutely essential. And so hearing about the challenges patients have even before there's drugs in development is very helpful to us, and helpful in knowing how we can even early on give advice to sponsors and guide sponsors. But I would just say that in all of these different sessions, the FDA staff listens very carefully. And I can't tell you how many times in discussions within our groups, as we look at study protocols, or we look at data, or we look at different aspects of a program, patients' comments are quoted or repeated. People are very much influenced by that. I can recall a number of instances where someone said, I didn't think this particular symptom was all that important to patients. That's really, this was really what we thought was really what patients were troubled by. But we heard at this session that this symptom really interferes with patients' lives and their ability to do the things they enjoy doing. And that really changes how we then look at the effect of a drug on that particular post-troubling symptom. Even in the design of trials, as I said, what we think the developer should be going after, how the endpoint should be crafted, is very much influenced by the input. And as Nicole mentioned, it's critical that the drug developers reach out to the patient community in developing the endpoints for trials. And we work to assure that they've done that what the trials are designed to evaluate reflects what patients think is important, reflects the patient experience of the disease, and that the drug can effectively impact that. So hearing from patients both about the experience with the disease, but also how they value the benefits that a drug is offering and how they would tolerate the risks does influence our decisions. But of course, we also have to focus on our responsibility to rigorously evaluate the data to be sure that the drug is providing benefit, that it is an effective drug, and that the benefit is outweighing the risk. And of course, in those determinations, we think about the patient voice, but we also have to be rigorous and do a thorough scientific review of the data in making our decision.
Thank you for that. I do think just realizing we're nearly at time and kind of going through some of the questions we've received from the audience, there does seem to be a lot of support for efforts towards transparency, but also kind of for future work or better understanding about how their initial feedback is being used in the process, and at what stage, and maybe where those difficult kind of discussions might have to be about where you're trying to balance various different evidence. So that seems to be most resonating with what we're hearing from those in the audience. Any last comments before we move on about what you really are working on or hope to see, but also just what you feel most hopeful for about better engagement with the patient communities moving forward?
Yeah, so thank you. I wanted to also highlight one thing that we hear a lot, and just to sort of underscore that we hear you, that in the gene therapy space, in rare diseases where there's just specifically not a lot of options for treatments, the patient community has told us we are willing to accept more risk to potentially get, benefit. And we hear that, and we certainly understand that. And so I think that I really want to underscore the fact that we are listening. I think that, in terms of just things to highlight or to end on, rare disease is a commitment, and the advancement of rare disease is a commitment from the Center for Biologics and also the Center for Drugs. I'm sure Dr. Stein would agree. And this is in particular an area that we are spending, I'd say, the majority of our time in trying to advance the gene therapy field and cell therapy field for rare diseases. We think that we have the momentum, we have the science, and we really would love to get to the finish line to support several of these rare diseases and to provide directed treatments that are really needed in this area. So I really do thank you for your engagement.
No, thanks. And I would just add to that we have been focusing on efforts to enhance and encourage the development of drugs for rare diseases. We started, as you started by saying, and as I underlined, there are too many diseases that lack effective therapies, and we are absolutely committed to finding treatments. We've launched last year the ARC program, Accelerating Rare Disease Cures program. We recently announced with CBER the START program. I think the GEMDAC will be an important addition to a way to review drugs. But I think it's important to say that in my experience, I've been in drug development for decades now. There's been a sea change, and it's certainly a sea change at FDA as well, which is, many decades ago, the concept of the patient voice was not as impactful or as central. It's absolutely central. Now we see a clear understanding of patients' needs as they tell us what their needs are, and their willingness to accept risk and the benefits that they're looking for as absolutely essential as we think about making decisions. We of course have to balance that with our responsibility of rigorously reviewing the scientific data to establish the drug does offer a benefit and that its benefit outweighs its risk. But the entire discussion includes is in the context of and is incredibly impacted by the patient voice and what patients tell us about their needs and what they're looking for. So we're very much committed to finding treatments for rare diseases and including the patient community in that process.
Thank you, Peter and Nicole. This has been valuable context on the current state of the agency's efforts, and in particular, how this meeting fits into the FDA's broader ongoing work to incorporate patient community voices. So I will next introduce Robin Bent, the Director of Patient-Focused Drug Development Program at the FDA's Center for Drug Evaluation and Research, and Dr. Teresa Baracchio, the Acting Director of the Office of Neuroscience at the FDA's Center for Drug Evaluation and Research, who will build on these remarks and provide more information on the drug review process and how early engagement can inform FDA's regulatory advice and decision-making.
Great. Good afternoon, everybody. So my name is Robin Bent, and as you just heard from Dr. Stein and Verdun, patient experience data is really critically important to the drug development process. And so we wanted to address a few questions that have been coming to us and that came up even more frequently from patient groups as we've been working to plan this meeting. Next slide, please. So I'm just going to take a few minutes to talk to you about some of the questions that relate to how we use the information from patient-focused drug development meetings and listening sessions. And I think a lot of this was touched on previously, but I just want to maybe provide a little bit more specifics. Next slide, please. So just at a high level, patient-focused drug development meetings give FDA, medical product developers, researchers, clinicians, patient groups, and others the opportunity to hear directly from patients and their loved ones about their experiences with a condition, as well as their unmet needs, treatment goals, and other important topics. Patient listening sessions provide similar information. They include fewer patients, are shorter in duration, and are focused mostly on sharing information with FDA. However, the summaries that are developed from the listening sessions that take place through the Office of Patient Affairs are made available to the public, as are the Voice of the Patient reports that come from the PFDD meetings. And so starting with the first question, how might the information from these meetings be used by medical product developers? FDA frequently hears from medical product developers that these meetings are very valuable, particularly when they're held early in the drug development process. When they're held early, these meetings really can be a starting point that provides valuable information to both FDA and the product developers about patient priorities, potential new targets for therapies, potential clinical trial endpoints, barriers to clinical trial participation, and other really critical information. And so just as an example, we've heard feedback from product developers. One person told us there were some areas and things that we heard about that were new for us and gave us the opportunity to do additional follow-up for a research question or finding that we had not thought about before. Another told us, the information that I heard at the PFDD meeting caused me to go back and make changes to the trial protocol that I'm working on. And a third person told us, by attending the meeting, we created relationships with the community to hopefully provide information about the issue and what we're doing to hopefully address the issue. And so now moving on to how FDA uses the information from PFDD meetings and listening sessions during discussions with sponsors. And that's really talking about throughout the drug development process. Our reviewers use the information gained from these meetings to inform discussions with the medical product developers on clinical trial designs and endpoint development. They also refer back to these reports as new questions arise. While I mentioned that several medical product developers are aware of PFDD meeting program and have attended the meetings, oftentimes FDA reviewers are able to refer sponsors to existing Voice of the Patient reports or listening session summaries. As well as encouraging the medical product developers to work with patients and patient groups to understand what is meaningful, particularly in the case of rare diseases. And I know that it's really, really frustrating to host a meeting and not immediately see results, and that for people who participate in the meeting, who are sharing really deeply personal stories and reliving really traumatic experiences in order to ensure that FDA and other stakeholders really understand what it's like for them and their loved ones to live with the condition, that to not know how FDA and others are using the information that they shared is really, really hard. But one of the challenges that we have is that we're not able to talk about exactly how we're using the information from a particular drug or from a particular meeting to inform our discussions with product developers. And so I am hoping that by sharing this information with you now, that you hear that this information is really important, and this information is not just going into a file to never be seen again. And let me just talk about later on in the drug development process when we're at the reviewing a marketing application. And so later in the medical product development process, the information that we get from the PFDD meetings and listening sessions can inform the benefit risk assessment by providing therapeutic context or an understanding of the impact of a condition on patients' daily lives, the symptoms that matter most to them, their experiences with available treatments, and their ongoing unmet medical needs. In addition, information about patients' willingness to accept side effects and uncertainties related to risk can be very important to review teams as they conduct their benefit risk assessments. And as the final question that I'm going to talk about today, it's if a PFDD meeting or listening session is held early in the drug development process and a marketing application is received by FDA sometime later, how can we be sure that reviewers, including new reviewers, are aware of the previously held meeting? And you just heard from Dr. Verdun that about the PFDD meeting that was held several years ago in sickle cell and how that did help to inform the recent approval. But also, hopefully, through answering some of the questions that today, I've been able to convey that the information that we hear at patient meetings is something that we refer to throughout our interaction with medical product developers. However, I also very much appreciate the concern, and given how much time and effort patients and their families put into sharing their experiences during the meeting, we want to be certain that reviewers are both aware that the meetings were held and that the information from the meeting is easily accessible to them. And so about 8 or 9 months ago, PFDD staff started reviewing the proposed indications for incoming marketing applications. And we checked to see if a PFDD meeting or listening session was held for the condition. If there was a meeting, we reach out to our review staff to ensure that they were aware that patient experience data exists for the application that they're reviewing. This information can be particularly helpful to new staff who may not have attended the PFDD meeting. Next slide, please. We also include a link to the website where they can find a list of meetings organized by disease or condition so that they're aware that the resources exist. We've gotten a great deal of positive feedback from our reviewers about our internal outreach efforts, and we plan to continue them. And I just wanted to share this screenshot of the webpage with you. It's public facing so that everyone is easily able to see what reports are available and access them. And I know from conversations with people working in the medical product development space that they also access this webpage quite frequently and refer their colleagues to it. Next page, please. Or next slide, please. And finally, on this slide, I put a few links about the pages that I mentioned previously, although if you type FDA and PFDD into a search engine in your browser, you'll be able to find all of the information. But if you do have recommendations on how we can make our website or others better, please don't hesitate to reach out. Next slide, please. And with that, it's my pleasure to turn the microphone over to Dr. Teresa Bracchio. Thank you.
Okay, can you go to the next slide, please? All right. So hi, I'm Teresa Bracchio. I'm the director of the Office of Neuroscience in Cedar, and I'm going to provide a high-level overview of the drug review process, but more specifically how patient engagement and stakeholder engagement fits into our review process. Next slide, please. Right, so have some disclosures on the screen. In addition to what's on the screen, I'll just note that the comments that I'll make reflect my experience in the Center of Drug Evaluation and Research. However, similar considerations also apply to the products evaluated in the Center for Biologics. Next slide, please. So there are a number of ways that patients and stakeholders can engage with the agency and share their perspectives in the drug review process. I've listed here some of our more common interactions, and you've just heard Dr. Bent refer to the patient listening sessions and the patient-focused drug development sessions. The patient listening sessions are held with the agency, and the patient-focused drug development meetings can be through the agency or also externally led meetings. In addition to these engagements, we also do have a number of other ways that we engage with stakeholders, such as through scientific meetings, working groups that are organized by different parties, public and private partnerships, and also research roundtables. In particular, we found a number of the roundtables meetings to be very helpful because they bring together industry, academia, government, patients, a variety of stakeholders to focus on a specific issue and really think about how to tackle that issue in new ways. These are just some common examples of interactions, but engagement is not limited to these settings. You can go to the next slide, please. So in this slide, I just want to highlight that although there's different stages of drug development that can be defined, stakeholder engagement really happens through all stages of drug development. The input can and does happen from the earliest stages in discovery and continues through post-approval. In the slides that follow, I will go through some of these stages more explicitly and discuss at a high level the activities that occur in the different stages and how patient and stakeholder engagement can contribute to development at each stage. Next slide, please. First, we'll talk about who makes up a review team in our group. And it's a multidisciplinary review team. There's a core review team that's made up of clinicians or clinical reviewers. We have non-clinical reviewers who are also referred to as pharmacology, toxicology, who review the animal studies that are conducted. We have clinical pharmacologists who review the pharmacokinetic or drug level data that is seen after administration of drug and help inform dosing. And then we have product quality reviewers who review the manufacturing and chemistry to make sure that good quality products are produced. And we also have statistical reviewers involved in the analysis plans and data that is collected in the studies. Depending on the needs at the different stages of development, there are other special groups within the agency that can serve as consultants for the review of a product, such as our clinical outcomes assessment groups who can advise on endpoints, and then pharmacovigilance and epidemiology and others as needed. You can go to the next slide. In the initial discovery and earliest stages of development, I've organized my slides in three ways to talk about what engagement happens at the level of the sponsor or the company that's developing the drug, FDA's involvement at this stage, and how patients and other stakeholders can be engaged. So initially in the discovery stage, that's when the company is usually doing their initial work to identify a lead compound that can target a disease. Small batches of the drug are manufactured, and they begin initial studies in animals to assess toxicity and inform dosing. In the very earliest stages, those are usually done by the sponsor without much engagement of the FDA very early on. However, after a lead compound is identified and sponsors are moving closer to doing studies in humans, they will often request meetings to meet with us where they can discuss the data that would be needed to support an IND submission and what the initial clinical studies that would be needed to support an IND. This is really a great time for initial engagement with companies and the FDA for patients to inform us about the understanding of the condition, and then what are the drug development needs. So it provides an opportunity. Often this is a great time for the patient listening sessions or the patient-focused drug development meetings, where sponsors and FDA can be educated by the patients about what the disease, what their lived experience with the disease is like, and also to reflect on, if we're going to move drug development forward in this space, what is needed to help support drug development? Is there a better understanding of the natural history of the disease that's needed? What are there outcome assessments that can be used to assess specific aspects of the disease? And also, it's a good time to mobilize the patient community to make sure that there are going to be patients available and willing to participate in clinical trials. Next slide, please. In the earliest stages, would be around the time that an IND is submitted, and initial clinical trials are done to first look at safety and tolerability, drug metabolism, and then for any early signs that the drug is having some effect, a desired effect, or targeting engaging the target that it's been developed for. At this stage, FDA conducts an initial review of the IND to ensure that studies do not place human subjects at an unacceptable risk. and we'll review protocols, safety reports, and things that are submitted to the IND. And we meet with sponsors and continue to meet with different stakeholders to provide advice on development issues. Under the patient section, the things that I've listed are not necessarily specific to that stage, but is sort of these things can happen at any stage, but it's just sort of examples of the type of engagement that would happen. Is that this is a good time to think more about what aspects of the disease are amenable to treatment, what should we target for endpoints. Patients can provide perspectives to further inform the drug development regarding benefit and risk, and specifically what would be a clinically meaningful benefit, how much uncertainty are they willing to accept on benefit, and how high the tolerance of risk is. And I can give a good example of a meeting recently that I engaged in that our group found very informative for drug development, which was it was a meeting. It was one of a patient listening sessions, and we had it was a neurodegenerative disease that had a marked impact on motor function, the ability to walk and balance. And we were aware of those aspects of the disease and the drug development that had been done up to that point had really focused and had endpoints that focused on things like walking and balance. But when we were talking to the patients, we also heard a lot about how their speech is impacted, their communication, and also that we heard from quite a few patients that pain was a specific aspect of their disease that had not previously been considered in clinical trials. And the people from my group who attended that meeting, when we walked away from that meeting, we were all collectively discussing, wow, those were some aspects of the disease we had never considered before. And we really have to, going forward, make sure that we are capturing those endpoints in the clinical trials in this disease. So we really do take these engagements to heart and learn from them and apply them to our conversations and advice that we give to sponsors. So next slide, please. Once we get to later stage of development, that's when we start talking about the design and conduct of studies that demonstrate safety and efficacy, and that could be used to support a marketing application. Sponsors are also conducting other activities at this stage to support marketing applications, such as additional manufacturing studies, non-clinical studies, and pharmacology studies. At this time, we have meetings with sponsors to reach agreement on study design and endpoints for these trials, and we discuss the ability of the data to support an application. And we continue to review protocols and statistical analysis plans and safety reports. At this stage, it's really crucial that patients can provide input on the elements of the trial design, such as the selection of study endpoints, and really importantly, the burden of assessments that are being proposed and the feasibility of the assessments. I think this is really critical input on whether patients are able and willing to participate with the number of clinical visits that are prescribed and the types of assessments that are done. Next slide, please. So this is not an exhaustive list, but here are some of the key considerations that we consider in trial designs. Each disease is unique, and each element of the trial design must be considered in light of the unique attributes of the disease. So when we think about what are the characteristics of the disease, and specifically, what are those that we actually think may stabilize or improve? What are the anticipated effects of the treatment, and what are the targets of the treatment? What do we think is reasonable to expect to see with treatment with the drug? We take into mind the size of the population, how rapidly the disease may progress, and how heterogeneous the disease is. And as I previously noted, it's very important for us to consider the feasibility of the study and the burden to the patients with the design of the study. Taking these considerations into account will inform the trial design, such as the trial duration, a selection of endpoints, the choice of a control group, the frequency of visits, and if there are things that can be done remotely or must be performed at a research site. Next slide, please. Patient stakeholder engagement, particularly in the rare disease space, has been critical for pushing drug development towards greater innovation. The overall goal with innovation is to increase trial efficiency and maximize contributions of patients. In rare diseases, we always carefully consider the need for a placebo arm, but with an effort to minimize trial duration and the number of patients. I've listed here some innovations that we are seeing more frequently and are working with sponsors to incorporate when appropriate into drug development. So external controls may be considered in certain situations. And earlier this year, we did issue a guidance for industry on external controls to help to define those situations where it may be appropriate and how to do so within a rigorous statistical framework. For the next one, seamless trial designs, because of the small patient populations, it can be challenging to follow what would be the usual order in a larger disease of drug development with the phase 1 study, then a phase 2, then a phase 3. In the rare disease space, we find that we frequently need to encourage the use of a seamless trial design, which combines multiple phases into a single study and maximizes the individual patient contributions across the development process. Platform trials allow for the investigation of multiple drugs in a single disease or a single drug across multiple diseases. This is most often useful in an earlier stage of studies to screen for potential benefit of products for further development. And one example of this in rare disease space is the use of the Healy platform trial that's being used for ALS. And that trial was initiated in 2020 and has showed rapid enrollment despite being initiated in the middle of the COVID pandemic. Decentralized trials not only make trial participation easier for patients, but it also allows expanded geographic outreach to patients who would not otherwise be able to participate in a trial. FDA has issued guidances on decentralized trial designs, and we are committed to expanding the use of decentralized trial designs while maintaining the quality and rigor of the clinical trial. And the development of more sensitive outcome measures that are capable of measuring clinically meaningful changes an example of this would be a digital health technology is also a priority for the agency. Next slide, please. This choice of study endpoints is a critical component of study design that benefits greatly from patient input. Ideally, we would like endpoints that measure clinically meaningful benefit in patients. However, in some situations, the use of a surrogate endpoint, such as a biomarker, may be the most efficient method to establish a treatment effect. A reasonably likely surrogate is a measure such as a biomarker that demonstrates an early change that is reasonably likely to predict a future benefit on a clinically meaningful outcome. However, once sufficient information is obtained to establish that the change does predict a future benefit, it is then considered a validated surrogate. In either situation, whether you're using a clinical outcome measure or a biomarker, the following characteristics are essential for selecting an endpoint that is most likely to be able to demonstrate a potential treatment benefit in a trial. And it's important that it is a concept or a marker that can be reliably measured, that it is anticipated to respond to treatment, and that it is able to demonstrate change during the duration of a trial. Next slide, please. Once trials are completed and suggest a potential benefit of the therapy, an NDA, new drug application, or a biologics licensing application, BLA, may be submitted. These applications include human and animal data and analyses of that data, as well as manufacturing information. An initial filing decision is made that by the agency that is based on the determination that the data is adequate and complete enough to support review. The review then takes into consideration all of the input that has been obtained from stakeholders during the drug review process, and Dr. Bent described that earlier about how some of the ways that we continue to incorporate the prior findings into our review. For approval, it is required that an application meets the requirements for substantial evidence of effectiveness and demonstrates safety that's for its intended use. As part of this assessment, we do conduct a benefit risk assessment that supports the use, and that benefit risk assessment must be favorable to support the use of the drug in the intended population. And I'll discuss that a bit further on a following slide. There are two different approval pathways that can be used, and this also depends on partly depends on the endpoint that's used. The traditional approval is based on a demonstrating substantial evidence of effectiveness on a direct measure of clinical benefit, such as how a patient feels, functions, or survives. Accelerated approval is reserved for serious diseases with unmet need, and this would be based on the use of a surrogate endpoint, which I discussed earlier, specifically a reasonably likely surrogate endpoint. And if that is used, then subsequent studies to confirm clinical benefit may be required. And in some cases, an advisory committee may be convened to advise on challenging issues. We don't need an advisory committee meeting for every application, but if there are issues that we encounter that we feel would merit further discussion and get advice from the council, then we may bring it to an advisory committee. And as Dr. Stein noted, this does contain an open public hearing session that allows patients to share their perspectives on the review. And we do find those perspectives to be very helpful in our decision making. Next slide, please. As I mentioned, we do conduct a benefit risk assessment as part of our drug review. And what that entails is that we have an evaluation of the demonstrated benefits and risks of the medical product. And then it entails making a judgment as to whether the expected benefits outweigh the potential risks associated with its expected use. An important aspect of this assessment is that it does take into account the therapeutic context, such as the seriousness of the disease, whether there are available therapies, and whether the drug presents a benefit over those available therapies, tolerance of risk, and the feasibility of conducting additional studies. And others, those are just examples. On to the next slide, please. So, and this is just a note that once a drug is approved, development doesn't stop there. There is ongoing surveillance that is conducted by both the sponsor and by the FDA of safety, and this requires the evaluation and submission of periodic safety reports. The sponsor will perform any necessary post-marketing requirements, such as safety studies or registries. And there's, depending on the mechanism of the drug, if it's applicable to other diseases, there may be potential for ongoing development in other indications. As for the FDA, we continue to advise the sponsor on ongoing development activities, and patients continue to have a role through continued engagement and input on ongoing post-marketing activities and voluntary reporting of adverse events, and providing any input on safety as they, during their use of the drug. Next slide. And this is the last slide. I'll just note that we do have a number of guidances that are relevant to the points that I've made in this in my discussion. And so these are publicly available, and you can go to our website and find these guidances that also touch on some of the same points that I've made. All right, thank you.
All right. Thank you, Robin and Teresa, for giving us that useful context. Now that we've laid down some foundation, I'd like to move us into our next session, which is engaging patients and other experts in trial design and related aspects of drug development. I'm happy to introduce the moderator for this session, Michelle Campbell, the Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience, Office of New Drugs at FDA's Center for Drug Evaluation and Research. I want to turn it now to Michelle to give us a brief presentation on trial design considerations, and then to moderate a panel discussion on the same topic. Michelle?
Well, thank you, Marianne, and I want to thank everyone for attending this afternoon. I know we have a very large audience and you've been putting some really great questions in the Q&A, and I do hope that we will be able to either answer your questions through the presentations and conversations we will be having this afternoon, as well as actually directly answering them through Q&A from these sessions. So that is just a little heads up to my panelists who are in this session, that there are some questions that have been posted by our audience that I'm hoping that we'll be able to talk to specifically in the next hour. I want to thank my FDA colleagues who started off this meeting, and especially to my colleague, Dr. Teresa Bracchio, who just went over a quick review of the drug review process and did talk about the trial design aspects and some things to consider. So next slide, please. And since I do not want to stifle a very informative conversation with our panelists, I just quickly want to highlight one of the slides that Dr. Bracchio discussed about considerations for clinical trial design. And I wanted to highlight a couple things that we think about from the agency, but also from the perspective of how can we make sure that all of our stakeholders as part of drug development are thinking about this as well as we approach clinical trial design. So first and foremost, we need to make sure we understand the patient population, the lived experience population of this rare disease or disorder. We do need to have understanding of what is the current prevalence of disease. Is this something that perhaps is a genetic-based disease that has recently been discovered with that mutation in a disease and was once before described as a different disease or disorder? And so how does that look, and what are those implications with those new findings? We need to have an understanding to the best of our abilities of disease progression. This is helpful of understanding, the onset of the disease. Is this a pediatric disease, or is this something that happens primarily in adults? How does this disease progress? Is this a slow progressing disease versus a fast progressing disease? Is this a disease or disorder that patients experience a regression of perhaps developmental milestones in the pediatric space, or loss of function? Or is it a delay in development of milestones and functional abilities? And so really having that understanding is critical, as that will and can inform us for optimizing clinical trial design and clinical trial duration. And finally, we know this is true for all of our rare diseases, is heterogeneity. And we hear this directly from the patient-focused drug development meetings and the patient listening sessions and research roundtable opportunities that we participate in. We hear directly about the heterogeneity of the lived experience in rare disease. And we need to make sure that we can account for that. I think the example that Dr. Bracchio gave was a really important one about our learnings from a recent engagement with patients of a neurodegenerative disease. And I think that is important. We know that heterogeneity is there, and we are always looking for ways to handle it. And there are ways that we can handle the heterogeneity and embrace that it's there. And we know that this is what makes a disease or disorder, and we know that those lived experiences are each unique. But understanding what does the heterogeneity look like, and how, and the impact of maybe disease and the progression is going to be extremely critical for that understanding. Next slide. And then when we talk about clinical trial design in a rare disease, is what can we do for that optimal trial design, right? We know because of the challenges of heterogeneity, of the rareness of finding an appropriate sample size, trying to get the most information with as reducing burden as much as possible and trying to find effective therapies. We really need to always be thinking from the very beginning with engagement with the FDA is how can we find that optimal trial design that will provide the most informative information that we would be able to review. So first and foremost, we need to make sure that the patient and caregiver lived experience has been taken into account, that industry and drug developers and medical product developers have taken into account that lived experience and the understanding of disease. At the same time, do we have that information as well on that lived experience. So Robin, Dr. Bent showed the internal mechanisms we have for us to see if we've had prior listening sessions and PFTD sessions. But if there's a specific topic or an area of a disease that we have some questions, we at the agency can also initiate opportunities to reach out to patient groups to learn more about the lived experience of a disease or disorder. So we also, from the other side, can do that reach out ourselves if we find it's needed. And I can say that I've been a part of that. And it is something that we definitely consider as an option if we need to learn more. We need to try to identify a selection of relevant endpoints that can support the clinical trial design and can support efficacy of a treatment. And what we need to make sure we understand is that we think these endpoints will have an anticipated effect on treatment, and that the characteristics of the disease is amenable to treatment, that we should be able to change those symptoms and that lived experience if we were to have a successful treatment. We need to make sure we address any safety concerns if we have treatments. Most of our treatments that we're hopeful for a rare disease will be lifelong therapy. We want to make sure that we've addressed any safety potential concerns that could develop over time. We also want to make sure that we've thought about the feasibility to enroll that population into the trial. And so that may be even a smaller population than the entire prevalence of that disease or disorder. Obviously, we want to try to enroll as many persons with that lived experience into our trials, but we need to make sure that it's feasible and that we are minimizing the patient burden for that participation. And that includes number of visits. It can also include assessments, laboratory testing that need to be done, to making sure that we are not overburdening the patient for their participation. And finally, we need to make sure that we're developing statistical analysis plans that can sufficiently evaluate the trial data. And this is why these conversations should be happening early. These examples that we've talked about are really why we do encourage the early and often communication with us to have these early conversations to think about it. But ultimately, we need to make sure that we can statistically be able to evaluate all the data that comes in from the clinical trial experience, from the patient lived experience, in a way that will allow us to be able to make a regulatory decision that we do have evidence of efficacy, and we have a benefit risk profile when we're looking at it that is safe for patients to be able to take that treatment. So with that, next slide, and actually you can go to the slide after that to introduce the panel. I would like to go ahead and ask my panelists to turn on their cameras and to begin our conversation about clinical trial design and thoughts about it. So we have today Daniel Boyce, who is a faculty member of the Informatics Core at Tufts University, School of Medicine's Clinical and Translational Science Institute. I have Emma D'Agostino, a patient and inpatient advocate for the Cystic Fibrosis Foundation. I have Thomas Miller, who's the Vice President and Global Head of Acute, Chronic, and Pediatric Disease Nucleus for Bayer. I have Joe Horgan, the Chief Medical Officer for AMO Pharma. Kelly Kidwell, Professor and Associate Chair of Academic Affairs of Biostatistics from the University of Michigan's School of Public Health. And then I have Rebecca Chu, our team lead and supervisory mathematical statistician from the Division of Biometrics 4 in the Center for Drug Evaluation and Research. So welcome everyone online, and thank you for joining us to be part of this panel today. Danielle, I want to start with you and see if you would like to open up our session with some open remarks and reflections of what we've heard so far today.
Sure. Yeah, I really appreciate, first of all, thank you very much for having me, and I appreciate being here. Yes, I'm on faculty at Tufts, but more importantly, I'm Charlie's mom. My Charlie is almost 14 years old, and he has a rare and catastrophic form of epilepsy called Lennox-Gastaut syndrome, and he started with something equally bad called infantile spasms. And so Charlie has really severe disabilities, neurodevelopmental challenges. And so I have participated in all kinds of things over the last 13 years, like many of the advocates and people with lived experience in our audience today, some of whom I recognize from some of my other projects. I have tried everything and went to everything, attended every meeting and sat on every panel. And so I've had that benefit of looking back. And one thing I just wanted to comment on is how things have changed over the last 10 years. Even with meetings like this, there's so much more openness and transparency around the challenges in the regulatory space, as well as the challenges from the patient. So that's one of the things that I really wanted to emphasize, the importance of us understanding each other and having a real deep dialogue and not a superficial 3 to 5 minute soundbite. And so what I've seen here, especially through my work in the ALS community, which is a beloved community, which also has a challenge with heterogeneity that you mentioned. I have seen, especially recently, we've seen issues with, patient advocates not understanding things like the statistical analysis plan and post hoc testing. And so really smart, well-meaning folks talking right past one another. And so I feel like this is a beginning of a really important dialogue around that, and trusting one another to understand the issues and the frameworks within where each of us operate. But as I was thinking about coming here and my participation here, I was, Charlie hasn't really been presented with a lot of clinical trials. Charlie is kind of far along in his journey. The inclusion criteria don't usually include him, if they even do include his disease, right? He's usually excluded from things. And so I've been thinking about how I'm the first person to donate his brain tissue when he had epilepsy surgery. I have the medical students in the room when we're in the hospital. I'm very open to this, but I don't know if a trial came up for Charlie. I don't know if I would volunteer, because of the challenges that we have in participation, and because I would have to be really convinced that it would be an outcome measure that would make it worthwhile to us. Like if they could stop his seizures so that every morning I'm not holding my breath to see if he's still alive, which is what all epilepsy parents do. For 13 years, I've done that. If you could make me not do that anymore, then maybe. But next week on Monday, he has a routine neurology visit. We love his clinic. That's not the problem. The problem is getting him there, taking him around. Are we going to have a nurse or a medical assistant who understands that he doesn't want to stand on the scale? Is someone going to ask me to sign something and I have to let go of him and he takes off because he's like an infant, but he's in a 13-year-old body? Is he going to have a seizure? Is he going to grab food out of some child's hand in the stroller? And so I think about layering in study procedures. And what I feel like is missing in so many of these dialogues is the understanding of the practical experiences and not just the sound bites. And that's where I really think we need to go deeper. And just really quickly, I've also been reflecting on some experiences I've had with sponsors that were really positive, where I felt like they really got us. And years ago, I won't mention the names, but years ago, I worked with a sponsor for our epilepsy community. And they would send folks from their team to come for days on end to our family conference. And by the end of that conference, we didn't need to advise them because they could instinctively understand what it was that would or wouldn't work. They would understand that it just makes no sense to make this formulation or ask us to do 12 hours of an all-day clinic visit, that we didn't have the money for a hotel room. And so they would need to pay that up front to us instead of reimburse us later, things like that. And so I feel like we really need to be more unvarnished with one another. And I think patient advocates, many of us are protective of our family or our dignity. I think sometimes we need to let that down a little bit in a trusted setting so that we can really, truly, all of us understand what it is that we're all going through toward the common goal, so that we're not talking past one another. And I think today is a really great start. So thank you again for having me.
Well, thank you, Danielle. Emma, I want to turn to you and see what and have you provide your opening remarks.
Thank you so much. I'm struck that I also was really thinking about the last 10 years. I'm really honored to be able to just share the story of CF. If you're unfamiliar with CF, it's a rare genetic disorder. And we have had a really exciting time in CF over the past decade. When I was born, we had, God is going to join this call. We really had an experience that is much more similar to a lot of the rare genetic disorders where we didn't have a lot of available therapies, and we've had a very transformative decade that has kind of paralleled, I think, the patient advocacy that we just heard about from Danielle. So we now have really transformative therapies, and those trials have happened as we've kind of had this genesis of patient advocacy. So we're in this new era of CF where we have these really incredible therapies. And now we have all this great research going on. But I think what we've learned with working alongside the CF Foundation and involving patients more in clinical trial design is it is so incredibly important, as we've heard this morning, to involve patients as early as possible in clinical trial design. If we can gather feedback early, it allows for really meaningful incorporation into study protocols, as opposed to just making patient involvement kind of a box checking exercise. We've really been working to involve diverse groups of patients. So getting a variety of perspectives, having patients from different socioeconomic backgrounds, different disease severities, getting that heterogeneity in CF. I know we're not the only disease where this is true. The patients where we're really struggling to recruit are the ones who really need to hear from. So we're working to make sure that trials are feasible for everyone. And I think that's a really important aspect of reaching out to patients. And we also in CF, as we heard this morning, a lot of talk about the sickle cell therapies that were just approved. We are also moving into genetic therapies, and that's another aspect where early patient involvement is going to be so important to make sure that those trials are going to be feasible, and also that we're clearly communicating with patients about what risk is going to be acceptable, and that we all are kind of on the same playing field to communicate what it means to be involved in a genetic therapy trial. So those are kind of some of the things that we're thinking about in the CF world, and I'm really looking forward to this discussion today.
Thank you, Emma, for your comments. And I think you've highlighted an important point about the difference between the gene therapy and drug trials, and what are those considerations that need to be taken into account, particularly with the emerging era of gene therapies for our genetic-based diseases. And that is look like a different conversation. And I think actually our next session may highlight some of that. Thomas, I want to turn to you and begin some thoughts from our industry colleagues, some of the remarks on what you've heard today, and thoughts about trial design and rare disease.
Thanks, Dr. Campbell, and thank you very much to colleagues at Duke Margolis and FDA for the opportunity to weigh in on a topic that's quite important to me. As you heard Dr. Campbell indicate in her introductory comments, I have a responsibility for the development portfolio for a relatively broad range of areas at Bayer, but of most importance to our conversation today, it's our pediatric development portfolio. And this has been an area of interest of mine for the almost 30 years at this point. And I could tell you, this has been an area that I've been intrigued with because of the challenges and complexities that are unique to developing drugs for children. But what really caught my attention as a younger man and as a trainee, how few drugs have actually been adequately studied for children. And this is even more especially true in the field of rare disease, where I spend a good bit of my professional time today. We currently have a portfolio that consists of small and large molecules, and more recently regenerative medicines as well, and agree with all the comments that have just been made in the last few minutes. The considerations with this new field amplify that complexity from a development perspective as well, from a number of reasons. But it's most so for children, where we're developing drugs in the pediatric patient populations present a number of unique challenges and have a number of unique considerations. First and foremost, children can't advocate for themselves. And this often requires that parents or caregivers have to weigh in their minds during the consent process whether they're comfortable allowing the child to be exposed to an experimental medication or potentially a control for an experimental medication. I have learned, maybe in some respects the hard way, but I am a firm believer that early engagement with patient groups is not only a nice to have, but a critical success factor for all the reasons that you just heard Danielle and Emma reference. We as drug developers love to measure a bunch of things, but what I've come to realize is that what sometimes we think is the right thing to measure, even after consultation with the health authority, may have little relevance to the caretaker or the parents. And so I find it as a good investment in time well spent to make sure that we consider these groups as part of the team from the beginning. I've also learned that different geographies oftentimes mean cultural differences with parents. We seldomly develop drugs for the United States alone. And I've learned that through, for example, the production of parent-facing materials, what would seem to members of my team, myself, as fairly straightforward and understandable, completely misses the boat in other parts of the world. So I think it's important to recognize that getting advice from parent organizations is important, but the representation from that audience needs to be as diverse as the geographies that you're considering in your trials as well. I've heard a number of reference to endpoints that are perceived as important as well, and I can tell you this is particularly one of the most particularly important topics that I engage in with parents. We, for some of the new genetic medicines, I know many colleagues in the ecosystem, I'm sure, have had many conversations about use of various wet or physiologic biomarkers, for example, as a means of testing whether the genetic medicine can bend the disease curve appropriately. But I find more often than not, these are not the type of things that parents are particularly interested in. What they are more often than not particularly interested in is activities of daily living. And how their child compares relative to their age-matched peers that are living a life without disease. And to what degree would it be possible, particularly with these potential new miracle medicines, to really dare to ask that question? To what degree might we be able to not only disease modify, but potentially even cure certain diseases? Time will certainly tell. The feasibility topic has come up quite a number of times, which I'm very glad to hear as a sponsor, because this is usually pretty high on my wall of worry in getting trials done in a timely fashion. Because in this particular case, when you're enrolling children in studies, you have to think about not only the individual and the inconvenience to normal life that it might have for the child, but also to the caretaker and the parent. So we have to think about things like the potential for missed work, for missed school, for disruption from after-school activities, if that's appropriate, so forth and so on. The trial has to fit into the ecosystem of the family, not the other way around. And I think this is important for health authorities to really dig into and understand as well, because a request for an additional study visit or 2 study visits can make or break the difference of feasibility for the child. It really thinking through the burden on the families as a unit, I think is an area that we can all do better with, for sure, everyone in the ecosystem. And then finally, on the design side, although I find this to be a particularly relevant topic in the United States. This is that parents and parent organizations have become much more sophisticated in the last decade and really probing on these topics. Traditionally controlled studies in rare disease populations are very difficult to do. And as the field of data science has matured, and some have alluded to this in prior comments, the ways of novel ways of controlling studies have matured as well, including approaches where we minimize and potentially even eliminate the need for prospectively enrolled control patients. So this is not just a clever way to design a study, but I think will directly impact to the feasibility of completing the study in a timely fashion. So with that, back to you, Dr. Campbell.
Oh, thank you, Dr. Meller. And those are some really great comments. And I'm already, just so our panelists, I'm already thinking about how to do the next question with incorporating what I've been hearing so far. So, Joe, I want to turn it over to you to get some of your thoughts and experiences when approaching clinical trial design in our rare disease space.
Yeah, and my story is a little bit unusual, or different maybe is a kinder word to use, in that my current job is I'm the chief medical officer at a small British American pharma biotech company, and we specialize in neurodevelopmental disorders and neuromuscular disorders. But my real story is I've actually played a number of been in a number of different roles that I think are relevant or germane to today's discussion. For example, I've consented my son to be, and I don't want to get into his medical story in this very large public presentation, but in a moment of crisis and anxiety and angst, I've been the consenting parent that converted him from a patient into becoming a research participant twice. And thankfully, the endeavor worked out well when all was said and done, but I will always, it'll always be a part of me, what that core feeling of emotional trauma was when I transitioned him from being a patient into being a research subject. And then subsequent to that, my career as this clinician scientist at UNC Chapel Hill, then at Duke. So I had the ability to be a sub-investigator, co-investigator, principal investigator. So I went through that process of not just taking care of patients, and my specialties in pediatrics. So it's typically parents, that I'm talking about, and the youngsters all have neurodevelopmental disorders, but not just looking after them, but also walking that journey with them as they're thinking through, do they themselves want to become research participants, and what are the considerations that they have to make? And in the instances of rare disorders, it gets quite challenging and tricky sometimes, because oftentimes the only way to gain access to the clinical experts is by becoming a research participant. And so if you join a research clinical trial, you can get into the tertiary clinic within 2 weeks versus 6 months, 9 months, something like that, if you just go through a regular conduit of patient care. And so there's a dilemma that produces itself there that I've lived through. And then in terms of being a principal investigator, wanting to be making a useful contribution to the endeavor, wanting to have good numbers, good metrics, which can sometimes distort, that perception of what's best for a patient, and in particular for a child. And then finally, my current role, where I'm charged with designing informative programs that are going to have informative readouts and that will keep untoward variability at a minimum so that the whole endeavor is a worthwhile endeavor. So all of these different roles that I've had, I think, have kind of come together to create what I hope is a higher level of empathy with regard to patients and caregivers and what their experience could be or will be when they make that transition from being patients into being subjects. And that's with me all the time, in each of these different roles that I've played, and in my current role as well. I want to make one last point, and then I'll sort of move it back to you, Michelle, which is in terms of heterogeneity. We've talked a lot about that. Multiple speakers have talked a lot about that today, and how it's an important aspect of, particularly in rare diseases. And one of the things I've been particularly interested in and keen to do when working with caregivers in particular, knowing what I study, is helping them to understand how crucial it is that if they are going to conjoin as being a participant in research, that they understand how crucial that it is to, for example, lean on observable findings versus subjective findings, and durable changes that may occur versus transitory changes, and then focusing on the heterogeneity again, the head to toe, symptomatology or expressions of the disorder. And they may not have that skill set to begin with because they've been living the experience of just their child or their youngster, with that rare disease. So that entrainment process, I think, to help parents and caregivers, in my experience, to become better research participants is an important part of my current endeavor, and it's something I'm really interested in. So back to you, Michelle.
Thank you, Joe. Kelly, I want to turn to you. I think you and Becky are going to be providing us, I think, a very important perspective of the statistical considerations that I think is important that we need to think about in this trial design conversation. I think we often are always talking about the endpoints, but once we've selected our endpoints, we have to be able to really measure them. We need to make sure we have good trial designs and what are appropriate number of participants do we need. So I want to get your thoughts on some of the efforts that you've been doing to really be looking at how can we maybe optimize clinical trial design in our rare disease populations.
Yes, thank you so much for having me and for having the biostatistician or statisticians role here. I think statisticians have a really important role to play in improving the design and analytic methods to improve treatment for individuals with a rare disease. As many statisticians are enamored with big data right now, we need more of us to be working on how to be efficient with small data. And in thinking about designs, as many have said already, we really want to think about how we can decrease the number of individuals on placebo, allow everyone to receive an active treatment, at least at some point in the trial, to allow those who are doing well to continue so that they don't have to cross over in an often used design, the crossover trial. We want to think about developing designs that can both identify and confirm a efficacious dose of the treatment in the same trial so that we're not having to do this multi-trial and longer term kind of phase system as we see in other diseases. We want to deal with heterogeneity as it's been talked about already. We also want to use rich registry or external control data, as has been mentioned as well, and figure out how we can provide robust conclusions from the small data that likely is going to mean that we need to move beyond the sort of standard frequentist framework and this p-value less than 0.05, which we kind of often think of as the norm. So if you go to the next slide, please, my colleagues and students and I have been really working in this space for the last 7 or more years and trying to develop designs, particularly what we're calling small sample SMART designs or small sample sequential multiple assignment randomized trial designs, where individuals can be randomized up to twice in a trial, and they are guaranteed to receive an active treatment or active dose of treatment. So this first slide that you see is a comparative effectiveness design where we're thinking about comparing 3 treatments that might be used in a rare disease, but it's not known which is best. This is a design that's currently being used. It's a trial in isolated skin vasculitis. And in the next slide, we've also developed designs to, so next slide, please. Yeah, there we go. We've also developed designs that are similar where individuals can be randomized up to twice to both find the best dose and also confirm that dose versus placebo. So while there is placebo included in this design, you'll notice that everyone who first receives placebo is re-randomized to receive either low or high dose, such that there will not be any patient that only receives placebo in this trial. We've developed Bayesian methods that incorporate that can incorporate external data. They also are using data across both of these stages to figure out what's the best first stage, either treatment or dose level. And the Bayesian methods provide really intuitive outcomes, like the probability of a treatment effect being clinically meaningful. And then, like I said, we can incorporate this external control or external natural history data. I will say that these designs aren't appropriate for all rare diseases. So these designs are appropriate for rare diseases that are stable or relatively stable over the 2 stages of the trial. So over the trial period, not stable for the amount of the disease. And it's also where the treatment's not curative. So it's likely not a good design for gene therapies. The designs, other researchers have also been developing Bayesian platform designs that address heterogeneity of patients, and they use historical data to model disease progression and produce individualized trajectories. We're also currently developing trial designs and methods that allow for multiple primary outcomes to account for heterogeneity of disease and the experience of that disease and the treatment effects. We really need more innovative methods and then dissemination and education to ensure that researchers, patients, and regulatory agencies really understand and see the advantages of these designs and the methods. I look forward to our continued conversation today because, as the many have been saying both in the chat and via talk, we have to engage patients early. I think it'd be really powerful to engage patients with statisticians to really push forward in the clinical trial innovation to figure out what do the patients need, what do they want, and how can the statisticians help get the robust findings at the end of the day. And then to include, obviously, the researchers and the regulatory agencies to ensure that we can improve the lives of those who live with rare disease. Thank you.
Thank you, Kelly. And Becky, I know you've heard a lot of great open remarks so far, and so I wanted to know, from the perspective of being a reviewer, statistical reviewer at the agency, and seeing a broad spectrum of rare disease sample sizes, trial designs, different methodologies, endpoint challenges. What are some thoughts that you want to open up with as we begin our discussion?
Yeah, thank you, Michelle. Always challenging to go last on a panel, particularly in this group. A lot of great comments that I've heard already. So like you said, I'm going to share a little bit just from my perspective, statistically, dealing with reviews here at the FDA. My experience specifically has been with the Division of Rare Diseases and Medical Genetics, which covers products for the treatment of rare inborn errors or metabolism. So when we're reviewing protocols, statistical analysis plans, and then later when the trial is completed, we're thinking about reviewing new drug applications for a rare disease. I think one of the most common statistical challenges is the lack of statistical power to draw a conclusion about the treatment effect. So most of us, I'm sure, are already aware, and I've seen some comments in the chat about the difficulties that just arise from having a small sample size due to the rarity of the disease and being able to recruit patients. But I think there are other factors that really exacerbate this challenge in the rare disease setting. For example, we're often faced with a lack of data to really inform what the expected effect size will be for the trial, or what would be even a meaningful effect size for patients. And that information is really key for statisticians to help us think about what sample sizes are actually needed when designing the trial. And that lack of information, it can occur for a few reasons. One can be due to lack of natural history studies. This can particularly occur if it's like a new diagnosis. Additionally, there's often limited information on the drug itself because we have limitations on performing those early stage studies like we might have in more common diseases. But because of this, there's often a reliance on a more optimistic effect size when we go into the design stage, and that results in a smaller sample size. The challenge there is that we may actually miss a more moderate but a meaningful observed effect when the trial is completed. And as we know, this is really difficult, particularly in the rare disease space, because we often only get one shot at these pivotal trials. So other factors that really impact power, Kelly has already touched on some of these, but the lack of sensitive clinical endpoints, heterogeneous clinical presentations within a disease has come up a few times, and the slowly progressive nature of a lot of our diseases, meaning that we really need to consider the trial length to make sure that it's long enough to detect an effect. But importantly, a lot of these challenges, I think, can be improved by leveraging patient caregiver engagement early on in the process. And I hope that we'll continue to discuss that today. But this information can really help us refine our design and analysis plan. And I like Kelly mentioned, statisticians speaking with patients, we don't usually have that opportunity. So we can incorporate kind of creative and innovative approaches like those that have already been mentioned. And that patient perspective, it really helps us understand the disease course, consider what we want to evaluate in the trial, endpoints as have already been mentioned, thinking about what a meaningful effect would look like and that we should be targeting, And then evaluating really what trial designs are going to be palatable to patients, that we can enroll and recruit patients for, and at the same time are considered ethical as well, and will give us that interpretable, reliable data. So I look forward to hearing and learning from all of you and looking through some of the questions today. And I'll hand it back to you.
Thank you, Becky. So I just want to let panelists know and then our audience know I'm looking at the time, and I'm going to say that we will probably only get to 2 big questions, but they're probably the biggest questions that we're seeing recurrent themes in our Q&A actually, of similar type questions. And they're actually kind of what we were thinking about when we met ahead of time to kind of discuss how do we want to have our conversation today. And so what I first want to start with is we've really heard some really great opening remarks about the importance of engagement, the considerations of populations as populations are aging, and what does that look like for trial design and information, the emergence now of gene therapies and cellular therapy options for some of our genetic-based diseases and that consideration. We're also thinking about, rare disease in itself has heterogeneity of what does a rare disease look like for that specific patient population, where we may have only 12 patients identified, worldwide, to do we have 100,000 or 1,000, that we know that trial design is going to look very different in what our capabilities are going to be based on what that prevalence level is. So and I think this is why engagement with our patients and caregivers for lived experience is really beneficial for all drug development stakeholders. And, FDA is one of those stakeholders, but as well as this esteemed panel is also part of that stakeholders and needing to learn from that. So I want to ask if you can think about, how can this engagement help when we are dealing with this varying size, perhaps in our rare disease development programs? And how is really having that information useful in designing that trial design, the feasibility aspect? We've talked about that. I have my own story of feasibility challenges. When I was a research coordinator, and it was definitely before patient-focused drug development, and I got that phone call from a parent saying, I think we want to end the trial because, it's going to interfere with school. And that wasn't taken into account. And had we talked to caregivers, we would have learned and probably thought about that a little differently. So I want to start with Danielle, if you have any thoughts, and then I definitely will come back over to the rest of our panel with some of those thoughts on how do we really balance what we're hearing from the lived experience with the trial design and the feasibility challenges, as well as how do I handle, particularly for Thomas and Joe and Becky, what happens when we definitely have to see this wide variety of sizes, and how those designs are going to look very different, and what we can do statistically will look very different. So Danielle?
Yeah, I just wanted to say I've been looking through the Q&A, and I just want to thank everyone for commenting, because as I said at the very beginning of my talk, I only represent me, and I'm only one disease, and heterogeneity is the word of the day, right? So we're all different. We all have different levels of risk that we're willing to take on, and different burden we're willing to take on. There were some comments about whether, I was anti-clinical trial, or as a group, we were not supportive of clinical trials. That's not at all what we're saying. I would do anything. For there to be a study for Charlie. What I'm saying is that Charlie would not be able to participate in many of these trials as they're set up right now without dropping out or missing visits. And that affects the power that we learned about later on by the statisticians. So without listening to the patient voice, which you're all doing here by sharing, these trials can be set up to fail. So I'm against non-patient centered trials. I'm certainly not, anti-clinical trial. And that's what we're here to talk about today, is how the patient voice can improve feasibility and recruitment and retention.
Thank you, Kelly. So Thomas, thoughts from you?
Yep, and maybe as someone who is definitely pro clinical trial, I particularly in children, because I think we have an obligation to protect children through research, not from research. But maybe two examples that relate to a couple of themes in the questions that you very nicely summarized, Michelle. On the topic of heterogeneity, I recall one example a number of years ago. We were first starting to think about a trial in pediatric chronic kidney disease. And as is often the case, we went out and had some exchange with the patient and parent community to get some feedback. And one of the areas of feedback that we received was, given what's at least the traditional endpoint for this disease in this patient population, to see a clinically meaningful change in that endpoint would often take several years in the adult equivalent patient population. And we heard very consistently from the parents that this was not acceptable, that they would not allow their child to participate in a placebo-controlled trial for that duration when new innovations may be emerging that they may be precluded from simultaneously participating in. It was a little bit of a circular dilemma that we had to navigate through. So back to the heterogeneity comment, one of the clever ideas that the team had at the time was wondering whether or not we could define a unique subset of patients for this particular endpoint in the all-comer population where the slope of decline was very shallow. Can we define a clinically identifiable population where that rate of change was much more rapid, what we referred to as the so-called rapid decliners? And the short answer is, using various large data sets, we were in fact able to define a rapid declining population. But derivatively, what that did for us, not only did it address the feedback that we were getting from the parent groups, but it intrinsically added a degree of homogeneity into our patient population, because we were looking now at a less heterogeneous subset of patients. So sometimes you can use feedback from patient organizations and some of more modern access to large data sets to overcome some of these conundrums. So that's example one. Example two, completely different program. We were looking at a rare complication of prematurity for one of our drugs that had theoretical application in, and we noticed that colleagues at another sponsored organization had a similar idea. And we asked ourselves, the relative time frame for the two programs was relatively contemporary. Inclusion-exclusion criteria, as we were thinking about it, was essentially identical. The question we started asking ourselves is, why couldn't we borrow the control population that was traditionally derived from the other sponsor's trial as a means of controlling our own trial? Essentially, could we use an external data set with a presumed high quality degree of data management that conform to all international guidelines as a means of controlling our own study, essentially synthetically controlling study? The point that I'll make quickly is the following. In my experience for doing this now for some time, the likelihood of attempted consent to secured consent in pediatric clinical trials is less than 50% in my real life experience. And we hear no more than yes. In this particular trial, that rate of secured consent was greater than 90%. And through no small contribution to the fact that all children that were participating in the trial, for the most part, had the potential to benefit from Verum if a benefit existed. So I think these alternative or increasingly utilized modalities or approaches in these rare disease studies have the potential to meaningfully accelerate labeling for new medicines for children in these type of populations. Not applicable everywhere, for sure, but in certain circumstances, potentially.
Thank you for that. Joe, do you have any thoughts from your experience with your vast experience in different roles and hats you've worn on how this information can help inform our trial designs?
Yeah, I mean, the word that comes to mind is fatigue. And so I'm very attentive to how fatiguing it can be to have an entire day when you're in the clinic, having to do all these clinical assessments that are on the schedule of assessments for a clinical trial. And much of the time as a sponsor, as a designer of clinical trials and being the individual that's responsible for executing the whole program, I find myself in the design stage just taking the black pen and the Sharpie and just cut that out, cut that out, cut that out, I don't want that. We're on our best day, our best assessment baseline and sort of end of study, the money shot, we're maybe going to get 2 hours of really on point attention, energy, good assessment. So anything after that is just going to be fraught with fatigue and untoward variability, which I also talked about, and the fidelity of it's going to just decline. So by the time that we get to those last assessments on the schedule of events table, we're going to see wider standard deviations are going to get blown out and what have you. So I'm constantly going through that process of trying to trim down the assessment to just sort of the core things. And again, kind of keep in mind, what is it like to be a caregiver, to be rolling through that process of a really long day, especially if there's things like a wheelchair involved or other, ambulation is an issue and what have you. So that's always kind of a key driver for me. And again, intersecting it with how fatigue can just degrade the viability and interpretability of outcome measures, because when they're packed in during that afternoon of that core end of treatment visit, that's again the money shot for change from baseline stuff, you can just get garbage, that's showing up that becomes uninterpretable. So it's just, trying to find that right balance is super important.
Becky, do you have any thoughts from a statistical perspective on?
Yeah, I think these are great points. So Thomas and Joe have made, Danielle. So I think in rare disease, creativity is huge. I think we've heard from a lot of people already that traditional approaches don't always work well, and so we need to kind of think a little bit more out of the box. In terms of your question about reducing patient burden, I think, having those early discussions with patients and identifying where that burden exists, what maybe it's fatigue in the case of Joe's discussion, and thinking through what we can do in terms of conducting the trial that would reduce that burden. This could be like reduced number of visits, reduced number of measurements at those visits. Some kind of direct data capture is sometimes feasible in certain settings that don't require clinic visits. This can also help improve kind of diversity in the trial, recruiting patients from other locations. Different options for follow-up when a patient stops treatment, considering how that's done, and consideration of different cultural needs as well. So I think thinking about those things up front, from a statistical perspective, what we're usually most keen on is getting that reliable and interpretable data. So the most we can do that kind of front loading about thinking what barriers to keep it, both having patients enter the trial and then keeping them in the trial, is really critical, especially in the rare disease space.
So thank you, Becky. So I'm going to shift to our next question, because I think this is going to probably take us up to the break. And I'm going to start with Emma and Kelly first. So you guys just know I'm going to come to you first, but I'm going to come to the entire group for this, because I think it ties in our entire conversation. And a reoccurring question that we are seeing in the Q&A is heterogeneity, but it's also heterogeneity not just in the lived experience, but it's how do I optimally try to select the concepts I want to measure to show success in a trial, right? And I think for, Becky and I as reviewers, we definitely see what comes into us, and we definitely see sometimes where we get like everything in the kitchen sink, and we're like, oh my gosh, patient burden and fatigue and everything that Joe and Thomas was head nodding as soon as he said it, thinks about going, this is just too much, right? What is the critical things that need to be taken care of? But in our rare disease populations, we know it's going to vary. We know we hear about this, we learn about this from our listening sessions. We hear you come and tell us about your varying lived experiences. And so from your perspectives, how do we really account for, and I think we've heard some really great innovative ideas already, actually, of how to take this into account, but how do we really balance the lived experience with the concepts we think we should measure, and how do I make sure that's the right thing to measure, because maybe my natural history is not really optimal at this time because we're still learning, on top of how do I make sure I'm going to have a success, and how do I design that, how to kind of analyze that? It's a big question. I feel like this is my everyday question that I sit and think about every day when something comes in. and we're faced with it. But I would love to get your thoughts, because I think this is really the crux of what we hear from patient groups and families is, are you sure we're measuring the right thing? You know, I think you're not measuring the right thing. We should be having this. It's the crux of how do we pull all this information together to tell that story of that experience, and really how do we utilize trial design, endpoint hierarchies to really be able to provide the interpretable data and describing of what that trial experience was. So when it comes into Becky and I and to our colleagues, we can be able to, you know, have be able to review the data. So Emma, you brought up some, also from your perspective, you brought up, you actually brought up some stuff with the gene therapies, which adds another layer, you know, of what that would look like. But what are your thoughts about for that you want to convey to us about how do we make sure we're accounting for that lived experience in CF? As you're right, it has evolved over time as new therapies have come along, and it is now shifted and it looks different than it did 10 years ago. But also making sure that we're still measuring the things that are important to you.
Yeah, so I think there's a lot of different directions we could go here, right? In CF, we've traditionally had a really set of criteria. It's been lung function, exacerbations, that's our endpoints, that's it. And we've also had a really set of inclusion and exclusion criteria, and it's pretty much been very standard for most trials. And over the past 10 years or so, as we've started to get patient feedback, we have started to question that. And as we've now moved into this new era of gene therapies and having highly effective therapies, where we are in a new era where we have a much more stable set of patients, where we have a higher level of health, that set of endpoints and that inclusion criteria doesn't necessarily make sense anymore. So a couple of things that we have done are, one, just started to question whether the inclusion criteria that we've always used makes sense at actually both ends of the spectrum. As you said, we always have to ask, if we change inclusion criteria, are we going to get the results that we want? So if we start to include patients that are at the healthier end of the spectrum, are we actually going to see improvement? Which is something that obviously sponsors always worry about, but can we look for opportunities where we can include the healthier patients and still think that we could see improvement? So that's one end of the spectrum that we can think about, is, do we really need to be capping lung function where we've always capped it for decades at this point? And then at the other end of the spectrum, we have always pretty much capped at the lower end of the disease severity spectrum and said, nope, if you have below 40% lung function, you absolutely cannot participate in pretty much any trial. And really, we know that patients with, say, 30% lung function are not necessarily at death's door. They can be quite stable. And there's not necessarily a reason to exclude them from trials. So our patient community has pushed very hard to expand at the lower end of the disease spectrum as well. And we've started to expand those criteria and really ask, is this going to ruin your trial if you expand out criteria? And I think being a little more inventive with criteria is one thing that we can do. Another thing that we've done, which is maybe a little more specific to CF, because CF is not just one disease mutation, it's many, but we've really parsed out our trials into different sets of disease mutations. And so we've run trials where in the initial trial, only certain people with certain mutations can participate, but the FDA has allowed us to go back and test in vitro to see if therapies could work for additional expanded populations, which is maybe a bit specific to CF, but just thinking about, are there other ways where we can include patients after therapies are approved, where maybe they couldn't participate in the trial initially, but can we get patients on therapies after the drug is approved, where maybe it doesn't make sense to include them in the trial initially, but can we be inventive about getting patients on drug even after the trials have occurred?
Well, thank you for that, Emma. Kelly, thoughts about approaches from a statistical perspective? I know there's been a lot of questions about some of your the method you were presenting, but what are some aspects from a statistical perspective that we may be able to take into account?
Yeah, so I think one of the important things is involving the statistician early, and then really focusing on, right, you asked about like, how do you think about the endpoints and think about the success of the study? It's through simulation, right? The statistician needs to be involved, needs to have understanding of the data that has been, whatever natural history or previous trials that are relevant, or electronic health medical record data exists, to try to use that to try to understand what, extrapolate to the trial setting, knowing that it's likely not perfect, but simulation after simulation, looking at different endpoints and or different decision rules, et cetera, to try to come up with the best optimal design, so that we can really hone in on, understanding from patients what are important, understanding from the data that we already have what seems to be important and clinically meaningful or improvable with treatment, and showing that to the regulatory agency to say, hey, look, this is what we think, this is how it could go under great ideal expectations and how it could go under not so great ideal expectations, and still how robust we think this trial can be and hopefully helpful for the individuals involved. So I think the statistician is really important to have the statistical underpinnings to be able to do that, but also to have really the clinical information and understanding to really know the science that's involved in this design, and its potential. So, I mentioned a few ways in which I think that heterogeneity can be incorporated in thinking about multiple outcomes or thinking about individual trajectories based on history data. There are lots of methods for which we can, try to dynamically borrow data and only do that where the groups are similar, such that we're not going to, hurt the individuals in the trial based on poor external data or based on borrowing from other groups in sort of a platform design. So I think there are methods there that we just need to ensure are well developed and well simulated and well understood to both researchers, patients, and the FDA. Thank you, Kelly.
Joe, any thoughts on what we've talked about and that question? I feel like for you, it's probably the everyday, as you've described, as you're taking the approach of how do I really design an optimal trial and implement it. But anything else you'd like to add to this conversation of how do we balance all of this?
Yeah, I mean, in my world, there's in terms of designing a clinical trial, doing a clinical program, that's the table. There's 4 legs to the table in terms of how we design it. And one would be clinician completed assessments or rating scales. The second would be, again, I work with children, so disabled children, so caregiver completed rating scales. The third would be functional or performance-based assessments. 10-meter walk, 6-minute walk would be an example of that. And then the last leg of the table is biomarkers, and that can be anything from DEXA scanning to plasma-based or CSF-based biomarkers. So when we're working with any stakeholder, whether it's a caregiver, an investigator, yourself, we're always talking about those 4 different legs to the table and what are we populating in the clinical study design that creates that leg to the table to create that solid multi-perspective assessment. And so in a simple word, it's composite. If we take a composite approach to assessment, and the scales themselves are composites, as you know, because if they're well designed, they'll do a sweep through the heterogeneity. But then that multi-perspective approach with the 4 legs to the table is in and of itself a composite assessment. And so becoming comfortable with that, including from a statistical perspective, becoming comfortable with that and understanding how that composite approach can be informative, I think is really, really important in rare diseases like what we've been talking about today.
Thank you. I'm going to turn to Becky, and then I'm going to go to Thomas. I'm going to end with Danielle. And so Becky, I think particularly highlighting some of the thoughts that Joe just mentioned of potential innovation and ways to approach endpoints. Again, you in the end will have to help as part of the review team review the data comes in and are part of the discussion we have in the early developments about approaches and how do we handle this. So what are your thoughts?
Yeah, great points so far from the panelists. So I think heterogeneity keeps coming up again and again, and it's a huge challenge in our programs. Having that patient engagement, which I know is the main focus of our discussion today, can really help us inform it how to prioritize certain endpoints. Usually we're thinking about a testing scheme and thinking about which endpoints, even if they are not the most important to every subject or every patient, that it may still be relevant across patients and we may be able to show a treatment effect in those. That kind of information is really helpful. I also agree with Kelly's comments about innovative approaches. We're very open to those in the FDA. Use of simulations can be really helpful to help us understand what the operating characteristics of those approaches are. The one thing I'll mention, there is a statistical area that I think shows some promise for addressing heterogeneity and kind of aligns with Joe's comments about using a composite approach, and that's the use of a global test. This is an approach that's actually been around for a while, but has recently gotten some traction in the rare disease setting. So a key advantage of this approach is that it can increase the power and show a statistically significant effect in situations where we have patients with different domains or different clinical presentations. The downside of this, and the reason why it may not be applicable in every setting, so it requires some discussion, is that it doesn't give us inference on independent endpoints. And a p-value may not necessarily indicate an overall benefit if we do see discordant results in those different endpoints. So, similar to multi-component endpoints or composite endpoints, which have been mentioned here, this approach usually needs to be supplemented with additional analyses to really understand the overall treatment effect. But I think again, creativity, thinking about these approaches that might be a little bit outside of our normal box will really help to move forward some of our rare disease programs.
Thank you, Becky. And Thomas, any thoughts you'd like to add to this conversation?
Yeah, sure. I guess the first one, thinking back to, I very much appreciated Kelly's comments. I find oftentimes that our study teams, the first time they're engaging with statisticians, they'll ask the question, what's the sample size? And I think that's the wrong question to ask. I think the right question to ask is, what's the option space? Help me understand the various ways that I have that we could apply rigor for sure, but and the implications of what that might mean from a study execution perspective. Because simply the selection of the design and the SAP, the statistical analysis plan and approach that's taken may have a non-trivial impact on time to labeling and time to patient. Number one. So that's guidance for my industry colleagues. On the heterogeneity topic, I very much appreciate both sides of the coin, right? Because in a perfect world, all of our drugs would be generalizable to everyone with a situation. But heterogeneity is not the friend of the drug developer, certainly in early development. Because if you can't ascertain a reproducible treatment effect under the best of circumstances, where you have as much sameness as possible, good luck with generalizability to a broader population. So I think it's prudent for everyone in the ecosystem to start, at least initially, with as homogeneous as a population as possible, understand what an order of magnitude of treatment effect would look like, to then subsequently inform your downstream studies. And then finally, on the topic of generalizability, in pediatric development programs, we always have obligations, but I think it's in everyone's best interest to have long-term follow-ups. And this is true, of course, for the new genetic medicines as well. And what we tend to measure in those long-term follow-ups often differs from what we were primarily focused on in our primary clinical trials. For example, in young children, we're often interested in neurodevelopmental outcomes. which were maybe not appropriate to measure in the short term. But now we want to have an understanding of is the child meeting their development milestones in a way that the state of the art would predict. So I think you could use your long-term follow-up to think about real life impact for these medicines well beyond the acute period during the context of the primary clinical trial.
Thank you. And Danielle, to end us, what are
Sure.
your thoughts from our reflection?
Yeah, so well, I'm kind of cursed because I'm a data scientist who specializes in real world data, but I'm also a mom. So when Charlie is, at clinic for a study, I'm thinking how noisy the data are going to be that day, and so, but I encourage the folks in the audience, the people from the patient communities, to challenge and to see the connection between feasibility and data quality and how this affects whether the study will come out the way that you hope it will. So instead of just saying, well, I'm just very lucky to be here, so we'll go through no matter what, no matter how difficult it is, we'll just do it. But sometimes that means that the outcome measures aren't measured the way they would be if you were not rushed, and all the other things that the panelists said today. So I just encourage you to keep asking questions about study design and data analytics. And this is very encouraging to me today to hear this conversation. It's one of the first times outside of my own meetings about this that I've actually seen this kind of engagement and answered this kind of questions. And keep asking those questions, patient advocates, because this is how we improve our studies and get good evidence, and hopefully someday find a cure for my Charlie and all of you as well.
Well, I want to thank everyone on this panel for participating today. It has been a great session, a lot of information in a quick time period. I want to thank the audience for putting some great questions in the Q&A. I wish we could get to all of them. They're really great, and I know that we will be taking them back at the FDA and looking at them. There are some really great questions. I, again, thank you all for this afternoon session, and I will just let us know that we will be returning. We're having a quick break. We will be back at 2:20 for our next session, which will be actually looking at case studies. Specifically, we have 3 case studies of engaging patients and other experts throughout the drug development and review process. So we'll see you back here at 2:20.
All right, it's 2:20. I know it was a short break, but we want to pack as much in with you all while we've got you as possible. I believe I've got everybody for this section ready to go. If folks could, if you're participating in this session, if you don't mind coming on camera, and I'm going to fire away. Welcome back. My name is Garrett Homry. I'm a research director here at Duke Margolis. I'll be moderating our next session, which is focused on case studies where patients and other experts throughout the drug development process have made significant contributions. We'll bring together patient community members, researchers, clinicians, and other subject matter experts for a few case studies on how stakeholder engagement patient group specifically, was critical in the drug development process. Then after 3 presentations, we're going to spend, similar to the last session, about 50 minutes with moderated Q&A and discussion. I have a couple things that were prepared based on the months of getting this meeting ready, but frankly, we have made lots of updates, both based on pre-submitted audience questions where we flagged a few things, and also we've been tracking the chat box of about 80 or so comments so far today. So know that you should be able to recognize incorporation of a lot of those points. On the next slide, I'd like to start by introducing our speakers for this session. As I mentioned, 3 presentations to start. First, Monica Morell, patient-focused statistical specialist within FDA's Center for Drug Evaluation and Research, followed by Jennifer Farmer, Chief Executive Officer at the Friedreich's Ataxia Research Alliance, and then Dr. John Sleesman, Professor of Pediatrics and Division Chief of the Division of Allergy and Immunology down here at Duke. John is also the co-director of the Duke Diagnostic Immunology Laboratory and medical director of Duke's Jeffrey Modell Diagnostic and Research Center for Primary Immune Deficiencies Diseases. After those presentations, as we're pivoting to moderated discussion and Q&A, we'll have a few more folks joining us. That includes Connie Lee, founder and CEO of the Alliance to Cure Cavernous Malformation. And then we'll also be joined by 2 folks from FDA, Dr. Lily Gerard a master scientist and technical lead of the patient-focused statistical sciences within FDA's Center for Drug Evaluation and Research. And finally, we've got Dr. Tejashree Purohit-Sheth, Division Director, Division of Clinical Evaluation, Pharmacology, Toxicology at FDA's Center for Biologics Evaluation and Research. Our first presentation will be from Monica, who will be presenting on PFIC and Alagille syndrome. Monica couldn't join us live today, so we pre-recorded her session. That's queued up. If we could go ahead and start that now.
Hello, I am Dr. Monica Morell, Statistical Reviewer and Psychometrician and Patient-Focused Statistical Scientist in the Division of Biometrics 3, CEDAR Office of Biostatistics. My presentation will discuss applications of patient-focused drug development in rare disease programs. The primary goal of patient-focused drug development is to better incorporate the patient's voice in medical product development and evaluation. To that end, FDA has published a series of 4 methodological guidance documents that address how stakeholders can collect and submit patient experience data for medical product development and regulatory decision-making. These guidances are available at the link at the bottom of the slide. Guidances 1 and 2 have been finalized, while guidances 3 and 4 are currently published in draft form. One way we incorporate patient voice in medical product development is through the assessment of whether an estimated treatment effect on an endpoint constructed using a clinical outcome assessment represents a meaningful benefit to patients. This can be especially challenging in the rare disease space, as it can be difficult to both find and administer appropriate clinical outcome assessments and to measure the patient's perspective on the meaningfulness of the treatment effect. Issues such as small patient populations, heterogeneous disease manifestations and progression make it more challenging to determine if a treatment effect is meaningful. Additionally, not all patients may be able to self-report due to cognitive deficiencies or age, and we may have to rely on caregiver or clinician reports, which do not directly capture the patient's experience. The anchor-based approach is a useful method for understanding what types of COA score differences are meaningful to patients. Importantly, anchor-based approaches are not one-size-fits-all, and the critical assumptions made by these approaches should be explored. What constitutes a good anchor measure will depend on the details of the medical product development program. However, some general attributes of good anchor measures are that they're plainly understood by respondents, sufficiently associated with the concept of interest captured in the COA-based endpoint, based on the same recall period as the COA-based endpoint, and thoughtfully developed and implemented. Additionally, non-anchor-based methods such as exit interviews can be used to aid in the determination of what types of COA score differences are meaningful to patients. As with anchor measures, the utility of exit interviews will depend on the quality and implementation of the interviews. The rest of this presentation will discuss 2 rare disease applications in which determining whether the treatment benefit was considered meaningful to patients was a review issue. NDA 215498, odevixibat, and NDA 214662, maralixibat. Both programs assess pruritus in pediatric populations. Pruritus is an itching sensation that creates a relentless and severely debilitating urge to scratch, which can lead to excoriation or skin mutilation. Given the patient's young ages, a single item OBS row was used to measure patient pruritus symptoms as observed by their caregivers twice daily, once in the morning and once in the evening. Additionally, patients who were considered old enough to self-report also completed an analogous single-item patient-reported outcome measure, which captured the patient's self-reported itching severity. These measures all utilized a 5-point ordinal response scale, with scores ranging from 0, indicating none or no pruritus, to 4, indicating very severe or worse pruritus. In both programs, the applicant conducted anchor-based analyses to aid in the interpretation of the pruritus endpoints. However, these analyses were limited by the quality of the anchor measures used or the implementation of the measures in the trials. As we'll discuss in more detail, the anchor measures either had no recall period or had a recall period that did not align with any of the applicant's pruritus pre-specified endpoints. These limitations presented a challenge for the review teams. How can we interpret the meaningfulness of the treatment benefit when there are no appropriate anchor measures? NDA 215498 for odevixibat was approved in 2021 for the treatment of pruritus in patients aged at least 3 months with PFIC. There was a single randomized double-blind pivotal trial with a sample size of 62. One review issue for this application was the assessment of the meaningfulness of the treatment benefit. Importantly, the interpretation of meaningful change depends on the specific endpoint definition, and there was a misalignment between all of the applicant's pre-specified pruritus endpoints and the recall period of the anchor measures. And while the applicant did conduct exit interviews, the way in which the questions were phrased and how the responses were recorded resulted in data that was not interpretable. During the review of the application, FDA proposed an additional endpoint to aid in the evaluation of clinically meaningful within-patient change in pruritus using anchor-based analyses. On the next slide, we'll look at how the review team arrived at that endpoint. FDA evaluated the comparability of morning and evening scratching scores within a given study day and score stability over time. This figure shows the degree of absolute agreement between patients' daytime and nighttime scratching scores on a given day between 4 weeks pre-randomization and the end of the blinded treatment period at week 52. We can see that during the 4 weeks immediately prior to randomization at day 0, there is only moderate agreement between morning and evening scores and a high level of day-to-day variability in scores. This suggests that morning and evening scratching severity notably differed during the 4 weeks immediately prior to randomization, and the degree to which the morning and evening scores differ on a given day varies notably over time. Given these findings, FDA proposed to construct the worst weekly scratching score over a 4-week interval as follows for each patient. The worst daily scratching score is computed each day as the worst of the morning and evening scratching scores for that day. Then a weekly scratching score is computed as the average of the worst daily scratching scores within the week. Finally, the worst weekly scratching score is computed as the worst of the weekly scratching scores within the 4-week interval to permit alignment with the 1-week recall period implied by the available anchor measure. Recall that observable pruritus scores were used to construct the focal efficacy endpoints considered by the agency in its evaluation of treatment benefit. However, given the first-time use of an observable to support evaluation of a drug product intended to treat a pediatric cholestatic liver disease, and that patients' itching severity is known only to the patients themselves, it was important to understand the relationship between scratching severity as observed by the caregiver and itching severity as reported by patients directly. To do so, the review team examined the comparability of worst weekly scratching scores from the observable, as shown on the last slide, on the y-axis, and worst weekly itching scores from the PRO on the x-axis, for the 9 patients who were old enough to self-report. Although inference is limited by the very small sample size, a high concordance was noted between these two scores, supporting the use of the observable scratching scores to evaluate treatment benefits in the pivotal trial. Figure 1 on the left shows the mean worst weekly scratching score in each treatment group for each month between baseline and the end of the blinded treatment period at month 6. Both doses of odevixibat reduced scratching severity on average relative to placebo, with clear sustained separation between treatment and control arms in mean worst weekly scratching score over time. The meaningfulness of this reduction in scratching severity was supported by the results of corresponding anchor-based analyses conducted by the agency. The figure on the right shows the empirical cumulative distribution of change in worst weekly scratching score from baseline to month 6 by treatment arm. We can see clear and largely consistent separation between the placebo arm on the one hand and the 2 treatment arms on the other, especially within the agency proposed range of thresholds for meaningful improvement in worst weekly scratching score, indicated by the dotted vertical lines at negative 2.14 to negative 1. Our second example is NDA 214662 for maralixibat, which was approved in 2021 for the treatment of cholestatic pruritus in patients with Alagille syndrome, 1 year of age and older. Trial 1, which provided key data for efficacy and safety, consisted of an 18-week open-label treatment period, followed by a 4-week randomized double-blind placebo-controlled withdrawal period. This trial had a sample size of 31. As with the previous example, it was important to understand the relationship between itching severity as reported by patients directly and scratching severity as observed by caregivers. Using the same methodology we saw 2 slides back, the review team found a high concordance between patients' self-report of itching severity and caregivers' report of scratching severity, which suggests caregiver-reported scratching severity may be considered an adequate representation of patient itching severity. The endpoint of interest was change in the weekly average of the worst daily pruritus severity score during the randomized withdrawal period, which was week 18 to week 22. The results, which appear in the table below, show a statistically significant difference between groups of negative 1.4. After the finding of a statistically significant treatment effect, the review team had to assess whether the treatment effect was considered meaningful to patients. While the applicant had submitted anchor-based analyses, interpretation of these analyses were limited, as the anchor measures included in the trial either had no recall period or had a recall period that was not specific to the endpoint, that is, to the randomized withdrawal period. The submitted anchor-based analyses were conducted at various time points and included data from the open label period, which can be biased, as respondents' knowledge of patients' treatment assignment is likely to influence how they report on patient pruritus severity. The challenge for the review team was to interpret the meaningfulness of the treatment benefit observed on the pruritus endpoint, given that there was no appropriate anchor data and no alternative data, such as exit interviews, to reflect the patient's perspective. This table summarizes the cumulative percentage of patients in the treatment and placebo arms experiencing worsening in pruritus during the randomized withdrawal period across 4 pruritus endpoints of interest: weekly average of worst daily pruritus scores, weekly average of daily, average, morning, and evening scores, weekly average of morning scores, and weekly average of evening scores. 100% of patients that were withdrawn from the study drug experienced a worsening in pruritus severity during the double-blind randomized withdrawal period, compared to between 54 and 69% of patients who remained on the study drug during the withdrawal period. FDA performed an exploratory descriptive analysis to examine the difference in change in worst daily scratching score between patients who continued on treatment and those who were withdrawn from treatment during the randomized withdrawal period. This figure displays the percentages of patients who experienced various levels of worsening in pruritus during the randomized withdrawal period. Starting from the left of the figure, approximately 54% of patients who remained on treatment during the randomized withdrawal period experienced worsening, compared to 100% of patients who were withdrawn from treatment. No patients in the treatment group experienced a worsening in the worst daily scratching score of greater than 1 point, whereas approximately 62% of patients in the placebo group experienced a worsening of 1 point or greater. What is not in this figure, but can be found in the review, is that approximately 29% of patients who remained on treatment experienced no change, compared to 0% of patients who were withdrawn from treatment. The review team concluded that given the large treatment effect observed across this and other pruritus endpoints, anchor-based analyses for the evaluation of clinically meaningful within-patient change in pruritus were not needed to interpret the treatment effect. The observed change in pruritus scores between week 18 and week 22 appears to represent a meaningful treatment benefit to patients and caregivers. I'll finish with some key takeaways from our experiences with these 2 applications. It's important to prioritize high-quality measurement in order to yield usable data. When all patients cannot self-report, it is critical to evaluate alignment between patient self-report and observer or clinician report. In our examples, a high concordance between patient-reported itching severity and caregiver-reported scratching severity bolstered the interpretability of the OBSRO-based scratching severity efficacy endpoints. These and other issues with the quality of measures used usually cannot be fixed once data has been collected. FDA exercised flexibility in evaluating whether the treatment benefits represented a meaningful improvement to patients in these rare disease trials. At the time of approval, there were no FDA-approved therapies for these diseases, making both areas of unmet medical need in rare disease with significant morbidity affecting children. We had high-quality measurements of an important symptom, pruritus, via caregiver report of patient scratching severity. And as we saw, both odevixibat and maralixibat were highly efficacious. However, it's important to note that highly efficacious treatment cannot always surmount substantive deficiencies in validity evidence. Finally, the use of exit interviews can be especially helpful in rare disease trials, but they must be thoughtfully conducted and responses should be captured verbatim, as data integrity is critical. Thank you.
And can we now go to Jen Farmer from Friedreich's ataxia for her case study?
Well, thank you, everyone. Garrett, thank you for inviting me to participate in the panel discussion today and to present a case study in Friedreich's ataxia. This is the story of a recently approved, the first approved treatment in Friedreich's ataxia. The drug is called omaveloxolone and now known as Skyclarys. My only disclosure is that this is Farah's version of the story. And there are no other financial interests for me to disclose. Next slide. So in terms of how this all started, there were several researchers who identified in several different animal models of FA a pathway that was paradoxically downregulated. This is an antioxidant response pathway called NRF2. And these were actually studies that were funded by Farah through our grant program. And, you know, I in the interest of time, I can't give the detailed version of the story, but next slide, you can imagine a mom, Ruth in this case, up late at night, Googling to learn as much as possible about FA when her son Jack had been diagnosed. And that late night Googling led her to those two papers. And Ruth's husband was a chemist who was helping pharmaceutical companies secure API. And Ruth put together that one of the companies, Reata Pharmaceuticals, that her husband was working with, was developing compounds that could activate the NRF2 pathway. And so Ruth put together that possibly Reata might have compounds that could be helpful in FA. And so the next morning she picked up the phone and she called Farah and explained to us her hypothesis. And the next thing that happened, next slide, is that Farah was able to reach out to Reata, and we flew down to Dallas, sat around the table with them, and explained to them that they might have compounds that could be helpful in our rare disease. Next slide. It took a little while to convince them Reata was developing these drugs for kidney disease. They were not in the neurodegenerative or rare disease space. And so, you know, it took a while for them to think about how these compounds could be useful in FA. Next slide. But they did recognize the opportunity, and the next thing we were able to do was facilitate a meeting between Reata, some of our key opinion leaders, our clinician researchers, and our community to think about how to approach clinical trials with these drugs in FA. Next slide. Next slide, please. So this kind of brings us to about 2015, and this was the chance to initiate the very first clinical trial. And so, you know, when we think about what the patient input was here, there was direct input in terms of clinical trial design. We helped Reata think about site selection. We had an ongoing natural history study. So we knew a lot about, you know, where patients were receiving both clinical care and participating in research studies. We were able to help with recruitment. And at the end of the part 1 study, we were also able to think about the interpretation of the results and help planning with the second study. Next slide. I'll just mention that this first study turned out to be much bigger than any of us ever thought it was going to be. We ended up needing 7 different dose cohorts. And so in the context of rare disease, this was a very large phase 2A study with 69 patients randomized 3-to-1. And it took us over a year to complete this study because of the need to go to so many dose cohorts. But it was important because it did show a dose response related to what became our primary outcome measure, which was a neurologic rating scale, the mFARS. Next slide. So that allowed us to move forward to a second study. Next slide. And this second study, I want to mention the importance of timing here too, was being designed and implemented in about 2017. And FARA had our externally led patient-focused drug development meeting in June of 2017. And I think that was really, it wasn't planned on purpose for those two things to coincide. But it was really helpful because we really needed to get alignment on the primary outcome measure for the study. And I think having that externally led patient-focused drug development meeting at that time allowed for both the sponsor and the FDA to work together with the patient community to gain confidence in that primary outcome measure. Our natural history study was also really informative in terms of designing the trial and understanding how many patients were going to be needed. And as everyone was talking about earlier, in terms of, you know, heterogeneity of these diseases, really being thoughtful about what the inclusion and exclusion criteria needed to be, because FA also has a fair amount of heterogeneity. And so we knew from prior trial experience that we needed to identify a group of individuals who where we were going to be able to assess this primary outcome measure in them successfully over a reasonable period of time, which in this case was 1 year. Next slide. So the outcome of the second study, and this was a double-blind randomized placebo-controlled trial, 1-to-1 placebo to treatment group over 48 weeks, showed a statistically significant improvement in the treatment group compared to the placebo group in this neurologic rating scale called the mFARS. Next slide. So this was really, exciting for our community. This was the first clinical trial that had a positive result. It was statistically significant. It was a robustly conducted trial. And Reata and FARA went to the FDA to review the clinical trial results and start the conversation of, does this meet the regulatory standards for consideration for an NDA? And this was, you know, I think as Dr. Stein mentioned earlier at the beginning of the workshop, you know, in the context of rare disease where there is no approved treatment, you know, but needing to apply flexibility, allowing for some element of uncertainty, but also still needing to meet substantial levels of effectiveness was kind of right where we found ourselves when we had the results of this trial. Next slide. And that really did lead to a process of regulatory engagement that FARA embarked upon in terms of trying to communicate the patient voice in this process, the patient experience, also making sure that data that was available could be shared and leveraged to help understand the meaningfulness of these results, and also encouraging, you know, the regulatory engagement between the sponsor and the FDA. Next slide. And so, as was discussed, I think earlier, there are different paths to demonstrating substantial evidence of efficacy, and there's more flexibility in the context of rare diseases. And so this was really the direction that the sponsor was trying to pursue, with a single, adequate, and well-controlled trial, and then identifying what could be confirmatory evidence to help support the approval of omaveloxolone in FA. Next slide. Next slide, please. During this process, the patient community was getting really concerned about the time it was taking to, you know, figure out what the next step was going to be. We were in the middle of the pandemic, and anxiety was just very, very high. And the patient community really wanted the opportunity to express their understanding of the results, what the results of these trials meant for them. And that resulted in a petition that was facilitated through FARA to RIATA and to the FDA to communicate the patient voice. And the clinician community really participated in this process, and also, you know, provided, I think, an important perspective in terms of consensus around understanding what the trial results meant in the context of the disease as well. Next slide. And, you know, the good outcome here is that we could come to, I think, a place where we could meet that regulatory standard. Next slide. RIATA and FDA did continue to meet. They did continue to discuss not only the results of the randomized placebo-controlled trial, but also the data that was coming forward from an open label extension study, and how to really build the case for confirmatory evidence from that open label extension study. And there are different ways in which you can try to, you know, where you can gather confirmatory evidence, and RIATA had worked to do that in a number of different ways in terms of understanding the mechanism of action of the drug in the context of the disease, in addition to analyzing that open label extension data. And the one of the studies that was done was a propensity matched analysis, where the individuals in the open label extension study were matched, highly matched to natural history control subjects. And that helped to provide additional confirmatory evidence that that eventually led to the approval of OMAV in FA. Next slide. And we received the approval of OMAV on February 28th of this year. And I should also add that this week, the EMA is also going to be making a decision on omaveloxolone in Friedreich's ataxia. Next slide. Maybe just one final comment now that we're kind of at this approved stage and we now have this treatment available to the patient community and people are now starting treatments. I was reflecting on some of the comments in the discussion earlier about heterogeneity again. And, you know, we could not have had a successful trial if we included all comers with FA. The numbers of individuals we would have needed would have been impossible for us to recruit and enroll. The duration of the trial would need to be very, very long. This was a homogeneous cohort of individuals that was enrolled in the study. But FDA, when they approved the drug and wrote the label, you know, did not restrict the use of Skyclarys based on the clinical trial. You know, the label is for individuals 16 and older, and it doesn't include anything about certain stages of disease or other characteristics. And I think that's really important. And I think going forward now in this post-market place, we can continue to gather evidence of efficacy and what's meaningful to patients through, you know, by seeing the experience that people will have who are at other stages of disease. And so, you know, those are, I think, additional opportunities that allow us to understand how these therapies can impact a whole disease population, while, you know, in the context of trials, we still need to be focused on a homogeneous cohort. So I think the last slide, please. Just a few takeaways. It's been, and I didn't have enough time to talk about our natural history data, but it's been incredibly important to our disease community to have high quality natural history data. And that's something that our community has been very dedicated to. And it's been invaluable to being able to conduct clinical trials and do so in a very informed way. Next slide, or just advance the slide for me, please, one more time. Thank you. Collaboration, I mentioned earlier, but I'm really grateful to both Reata and FDA for the continued collaboration and commitment to evaluating the data and working together to apply flexibility and, you know, make sure that this, for this program, that we were able to meet the substantial level of effectiveness criteria. And communication among all stakeholders, especially with the patient community. Thank you for your attention.
Thank you so much, Jen. And then one more case study from Dr. John Sleesman from Duke, who will be presenting on congenital athymia. John? Oh, I think you're still muted, John.
Thanks, Garrett, for inviting me, and thank you for allowing me to talk about my journey and our Duke's journey in the development of treatment for congenital athymia. Just my disclosures, Duke does receive royalties from Sumitomo Pharmaceutical, which currently owns the patent for the BLA that was associated with the development of what's now called Rethymic, which is used in the treatment of congenital athymia. Next slide, please. So this is a very different type of paradigm in that this is a syndromic condition. Congenital athymia is a syndromic condition that is the result of an embryopathy that occurs very early in fetal development between the 3rd and 4th weeks of gestation. And what happens is that there's a cellular disruption of the formation of the brachial arches, which forms the conal truncal parts of the heart, the thymus, the parathyroid gland, and also causes dysmorphic faces and dysmorphic anatomy in the children that are born with this condition. In addition to this, multiple genetic conditions can result in athymia. For example, 22q11 deletion syndrome, which is a very common deletion, one of the most common deletion in human genetics, and it affects about 1 in 3,000 babies per year. Yet congenital athymia in association with 22q deletion syndrome only occurs in about 1%. So the spectrum of the clinical condition is broad across the different genes and embryopathies that are associated with, including diabetic embryopathies, a gene called PAX1, another gene called FOXN1. So we have multiple causes for the same syndrome. The other thing that's confusing to both physicians as well as to families is that we're only addressing the treatment of one of these syndromic conditions, that is the loss of the thymus. The thymus is responsible for the development of T-cell immunity, which directs the adaptive immune response. So these babies are born without T-cells, which makes them susceptible to autoimmunity as well as to infections. Yet treating the thymic aplasia does nothing to any of the other syndromic features. So parents and families are often confused that somehow treating the athymia will also fix the other syndromic features of the disorder. Next slide, please. So if we look at the journey, if you will, for the licensure of use of thymic tissue implantation to treat congenital athymia, the first step was really a viable way to culture human thymus ex vivo. And interestingly, that was actually funded by HIV research in the late and mid 1990s. At Duke, this was funding through Bart Haynes' studies of human thymic development. And as a result, the idea came up from Louise Marker to use cultured thymic tissue to treat children that were born with no thymic tissue. And she had a proof of principle study that had 5 children that were treated successfully at Duke between 1997 and 1999. This led to support from NIAID, but more importantly, support from the FDA to do cost recovery in order to help pay for the cost of getting children to Duke for treatment from all over the country. This program would not have been possible without that cooperation between Duke athymia program, the FDA, and third-party payers who were willing to use what was extensively an experimental treatment, but was life-saving for children that were born with athymia. And Duke continued to provide support throughout this time. And pharma was really a late comer. It wasn't until 1918 until we got pharma support in order to carry this through FDA approval for allogeneic processed thymic tissue in October of 2021 under the auspices of the 105 children that were enrolled in the pivotal trial going back to 1999. The other key time point that really affected this program was 2018. Between 2011 and 2018, newborn screening for severe combined immune deficiency was implemented in all 50 states in the United States. And now we were beginning to diagnose these children at the time of birth, and they could be referred for definitive treatment. So many of these children who had athymia were not recognized because there was no mechanism to screen them until they developed severe infections or until they developed severe autoimmune disease. Next slide. Next slide, please. Oh, one too many. Oh, back one. One more. Thank you. Okay, great. So if we think about the clinical outcomes of children that are born without a thymus, T cell development really begins in the bone marrow, and cells migrate to the thymus, which is located in close proximity to the heart. And these cells undergo the ability to recognize not only antigens, but also to recognize self. So the T cells that come out of the thymus, which are signified in the little blue circles here, do not develop at all. So these children have no T cells, therefore they have no cellular immunity to protect them from severe infections. In addition to that, some cells are able to undergo aberrant T cell activation and differentiation, and they result in autoreactive T cells, and over 40% of children with this condition develop severe autoimmune disease, which is a major cause of morbidity and mortality in these kids. Next slide. So if we look at the natural history, and this is a really important aspect of the treatment of athymia, the children, the 105 or 95 children that were included in the pivotal trial, we had about a 20% loss to morbidity and mortality over the first year, but after that, that Kaplan-Meier graph remained relatively constant over the follow-up period. However, those children that did not receive treatment, most of those children died before age 3. The caveat to this particular slide is that this is data that goes back almost 20 years and before the advent of newborn screening. So we have to reset how we define the natural history, because we are able to implement treatments ahead of time for prophylaxis, which I'll show you on the next slide. So now with newborn screening, we're able to put these kids into protective isolation, initiate prophylaxis, monitor the development of autologous graft versus host disease before they received thymic implantation, and then we monitor their T cell reconstitution after that. Next slide. So the actual process of giving thymic implant, the thymus tissue comes from a child who is undergoing elective heart surgery. The pediatric heart is about the size of a walnut, and the thymus in this child is about is about the size of a man's hand. That tissue is removed at the time of elective heart surgery and otherwise would be discarded. But in this case, we're taking it out, culturing it ex vivo, actually removing the donor T cells, having an intact thymus stroma, and then giving that back to the child who was born without a thymus. Next slide. So the way this works is after the 2 weeks of culture, time in culture, there's a small incision made in the quadriceps muscle. The thymic slices are placed into the quadriceps, and then the skin is closed, and we wait for the T cells to develop. The reason we put it into the quadriceps is that it's easily accessible, so it's a minor surgery. And in addition, there's high vascularity in this region, which provides the blood supply to maintain thymic development. Next slide. Next slide, please. So after about 3 to 4 months, we see signs of normal thymic tissue, as you can see in the thymus allograft at 3 months after implantation. And using our biomarkers of reconstitution of naive T cells, we can see emergence of intact T cells, usually between 12 and 18 months post-implantation. Next slide. Right now, one of our biggest problems are the fact that this treatment is only available in 2 locations in the United States, here in Durham at Duke, and also in Great Ormond Street in the UK. And the cost of this therapy is quite high right now, $2.7 million in the US for the finished product. Next slide. So Louise Markert has spent her entire career in developing this program at Duke with the help of multiple collaborators here. We have received a great deal of support from both the Clinical Immunology Society and the the Pediatric Primary Immune Deficiency Transplant Consortium, and they've been our primary partners in referrals and getting patients that are identified through newborn screening to Duke for referral and treatment. Thank you.
Thanks so much, John. Now, as we start to turn to the discussion phase, Connie, I'd like to start with you to provide some additions or reflections following these presentations. And if I could tee it up with the fact that, listen, you've been involved with this type of patient advocacy work and in the difficult rare disease space for decades, really, and something that you, me, and Jen talked about as we were preparing for this session was that there's a lot of great stuff to tie in from John and Jen's case studies and also Emma's earlier with CF Foundation. But ultimately, in the end, they were successful. But as Peter highlighted at the start of the day, that's not in the majority of cases with rare diseases. We don't have therapies. This is really, really difficult stuff. Could you start off, though, by sharing a few minutes about your work and about any insights, and frankly, from anybody in this regard, about thoughtful suggestions to patient groups that may not have had kind of this level of success yet? Connie, will you please introduce yourself, and then just to give other folks a set of an idea of where we're going with. After your response, Connie, I do want to make sure that the two FDA folks on the line kind of briefly introduce themselves and talk about their work very, very briefly, just so that folks have context, because later on we will have a couple questions that are very FDA specific that would have been coming through the chat a lot, trial designs and other approaches that work for very small, often you hear the word, the vernacular, ultra rare diseases. So definitely want to ask that. Also want to talk to really everybody. Natural histories component is so critical. And as much as we can talk about the how registries were leveraged, how patient groups were utilized in developing these natural histories, and how ultimately these natural histories, as we can see again and again, came to have such an important role. So so probably the next 15-20 minutes or so. But Connie, can we please start with you?
Sure. And I have slides, and we're going to do the slides, but I think there might be a little bit off slide talk as I've been listening to the day, and I'm really hoping to be able to ground this talk a lot in the practicalities of running a patient organization and what the possibilities are and the opportunities of running a patient organization. Next slide, please. So just the teeny bit of background, cavernous malformations, the disease that I work with, also known as CCM, are abnormal blood vessels in the brain and spinal cord vasculature. They look sort of like raspberries or mulberries. They're in the capillaries, so blood flows through them relatively slowly. What can happen is they can hemorrhage. In about 25% of cases, people who become symptomatic have hemorrhage with resulting stroke symptoms. Sometimes those resolve, sometimes they don't, and they lead to disability. You can have seizures, you can have functional neurological deficits as a result of growth of these lesions. They can become 4 or 5 centimeters large. There's a sporadic form, there's an autosomal dominant familial form, and currently we don't have a drug. Our only treatment is brain surgery, and that's not always possible. Next slide. So we have worked with a number of industry partners, but there's two that I want to focus on, and I specifically, because the listening today, want to focus on the FDA meetings that we've had. Our first industry partner we brought in, much as Jen mentioned, this is a role that patient advocacy can play. We were frustrated in not being able to bring in a Japanese company who had developed a drug that was successful in mouse models for our disease. And so we Googled to find other companies in the US who might be developing something similar, and we emailed them, cold emails. One of them responded and was interested. They were developing the product for something different, and they were able through us to connect with our mouse researchers and start doing the preclinical testing that led them on. We consulted on the patient journey, on trial design, and then we recognized that what we needed to do was figure out endpoints. We had a lot of natural history data. We we know that hemorrhage is not a common occurrence. It's infrequent events, and that's problematic in clinical trials. So we requested and organized a CPIM meeting, Critical Path Innovation Meeting, and specifically bringing in 2 different industry partners, our Scientific Advisory Board and us, to talk with FDA in one big sort of intimidating room on the campus about what the potentials were other than hemorrhage. Could we do imaging biomarkers? Could we do a patient-reported outcomes measure? A lot is riding on this. And at the end of the meeting, FDA said, nah, you're doing hemorrhage. There's hemorrhage enough for you guys to do that. And all of us walked out feeling gut punched, like this was going to be the end of us ever having the opportunity to have any sort of treatment for our disease, because we had shown them the statistics that just really, for a cost possible trial, that was not going to be an option. And you see my little line there that says that this drug is now in phase 1. So you will hear how we kind of got around that or started to solve it. The fatal error that we made with this CPIM is having it before the patient listening session. FDA had no clue about our disease. We walked in there, we shoved a bunch of numbers and pictures in their faces, and that was all they knew. They had no background in patient experience. So when we got to our next drug company, one of the first things we did was organize a patient listening session. I've seen comments here in the Q&A. It was free. CPIM was free. We just had to get there. We didn't have a consultant. We didn't do anything. We showed up with our patients and their stories in time-constrained ways. We shared that hemorrhage is not the only issue with our disease. It's a recurring and remitting course that our disease takes. It's very much more similar to multiple sclerosis. That opened the doors. So our second partner, and actually going back to our first partner now, are able to have inclusion and exclusion criteria that much more match what the patients want, the disease course itself, the endpoints that we're looking for. We're still in phase 2 right now, so we're still looking at safety as an endpoint, but the exploratory endpoints all are the result of the listening session that we had. In the meantime, we've also participated in Type B and Type C FDA meetings, and every time we go, we tell the patient story and we reflect back on that listening session and the things. The listening session was also held on the campus in a conference room that was standing room only, plus a whole bunch of people on listening in from the outside. They had a little speaker that you could, it could be picked up on. So with our second drug company, we have done the full soup to nuts that some of you have already experienced, getting them trial sites, introducing them to PIs, CRO and staff education, recruiting, development of now a patient-reported outcomes measure that they're using. Next slide. And while that slide is turning, their phase 2 enrollment completed ahead of schedule because of the work that we did. So what's our role as patient advocates? We have a role to play in preclinical research. For us now, we're to the point where we are establishing our own mouse colony so that industry can come to us and start testing their drugs rather than having to work with academia. We have a complicated breeding scheme to get to our mice, so our experimental mice, so we can do that. We're small. We're a 1/12 the size of Farah. I just checked. And so we're not huge. We're just focused. We can partner in regulatory, as I mentioned, by timing them well. Make sure you time them strategically. We can be full team members in the clinical side, as I mentioned. Okay, now here are my cautions, and then I will be quiet. Not all diseases have patient organizations that have scientific expertise. Our second employee was our chief scientific officer, and I would encourage everyone to bring science in early. Not everybody has the connections. Reach out to your scientists, start those meetings. even mature PAGs have varying strengths. And I think it's on industry to evaluate the PAGs in your space to see how they can best help you. Not everybody's going to be able to help you in the same way. And that might be a role for FDA to help with these maturity and strength assessments. And then the last thing is about money. Working with a patient advocacy group without a service agreement on the part of industry is not in either party's best interest. I firmly believe this, having done it both ways now. And I don't think it's in regulatory's best interest either. And so I'll hand it back to you, Garrett.
Okay. So many great specific insights there, Connie. Thank you so much. Before I jump to introducing, to asking the FDA folks to introduce themselves, John, Jen, anything that stands out from the items that Connie shared? So many kind of prescriptive, specific experiences. I don't know if you all have any responses that you would like to share.
Follow up on Connie's comment about funding and support, because I think that that's critical for us. Pharma came in very late in the development, and much of the heavy lifting fell on either support from NIH or collaborations with the FDA for the kind of overseeing the cost agreements that we needed to put in place to bring these complex kids in from all over the country. And without that defined financial underpinning, it wouldn't have been possible to do it at all. So I think that that's an area that could really be developed more. And it's in the best interest of getting the approvals through quicker.
Flagging resources and industry to go back to later in the conversation. Thank you. Jen?
Yeah, I just want to reiterate a point Connie made that, when we started down this whole process, or even before that, FARE started as an all-volunteer organization with a scientific, 3 scientists and patient families. And when we engaged with Reata, we were a very small organization when we got started. And, yes, we've grown over time, but, a lot of the things that were done in the past that helped get us to where we are today, they all started with limited resources, and you just continually slowly build and build and build, and so I would never want, a story like ours where we are today to intimidate anyone from starting the process. And much of the things that we started early, and many of the things that we do on the engagement side are not high lifts in terms of cost, as Connie mentioned. These things can be done very efficiently, and they still are today. So I'm really glad you brought that up, Connie.
Yeah. Okay. Thanks to the 3 of you. We're going to get back to you in just a minute. Lily, Joshua, could you please briefly introduce yourselves and talk a little bit about your work, so that I can, when I share a couple questions related to what we talked about at the start of the session, folks have a context as to why we were so excited, kind of adamant to bring you into this conversation.
Okay, sure. I can start. Hi, good afternoon, everyone. My name is Tejashree Schatz, and I am the Division Director for the Division of Clinical Evaluation General Medicine in our newly formed Office of Therapeutic Products in CBER. I have been with the FDA for over 21 years. I have pre-market review experience in CDER, CDRH, as well as CBER. I have been here close to 7 years now, overseeing product development, particularly for rare diseases with cell and gene therapy. And so, very happy to be here, and really appreciate this opportunity to participate as a panelist in this very important conference. We sincerely appreciate the significant unmet need in rare diseases. And as you have heard throughout this conference today from our FDA colleagues, we are really committed to support product development for rare disorders. Additionally, as you have heard, patient input is extremely important to FDA in our regulatory oversight of medical product development programs throughout the whole product development lifecycle. As sponsors identify a target condition for their product development, it is really important to engage patients early to identify what is really important to the patients. And with respect to the disease, the symptoms that are important to them, those that are bothersome to them, the impact of the disease on the patients, what their perspectives on available treatment, what constitutes an unmet need for them currently, and also to understand the natural history. As you've heard, natural history data collection is really important. And then also to get an understanding of the patient's perspective on the benefit risk assessment, right? I was on a panel a couple of weeks ago, and I heard from one of the patient groups, and this was really, really important. They said, we are willing to accept a large degree of uncertainty, but it depends on what the benefit risk trade-off is. So large degree of risk for large degree of benefit, small degree of risk for small degree of benefit. So, I think, hearing some of these is really important, and understanding from the patients for that particular condition, what they consider as benefit risk trade-offs and degree of uncertainty they are willing to accept with respect to benefit risk. And collecting natural history is really important to us, and honestly can serve as a external control, particularly in the rare disease space for single arm trials. And the patient information that is collected can inform, as you've heard, the design of clinical studies, particularly the first in human studies. A lot of times, our stakeholders are looking for patient input for design of a pivotal study per se. But even for early phase studies, the information can be really helpful to identify critical study design elements, to include potential safety and efficacy endpoints, study assessments, and burdens of the required assessments, to include timing, frequency of these assessments. We mentioned risk tolerance, acceptability of risk mitigation, etc. And in the gene therapy space, particularly for these ultra rare diseases, the first in human study may actually be the study that may be provided to support licensure, dependent obviously upon the outcome of the study and the results that are provided to the agency. So it is really important to collect meaningful information on benefit, even in these early phase studies. And you've heard that patient experience has been gleaned in the context of patient-focused drug development meetings, patient listening sessions. We've also found patient engagement helpful in the form of patient representatives participating as special government employees that provide and address some questions in the review of our regulatory submissions. Patient experience feedback in the context of advisory committee meetings for our marketing applications. We receive sometimes hundreds, if not thousands, of written comments that we review and seriously take into consideration, as well as comments we hear in the open public hearing. Patient engagement workshops are also helpful, and we also want to encourage our sponsors to bring patient representatives to our regulatory meetings with our sponsors. We find that very useful. So I think that patient input is critical throughout product development, and we look forward to working with our patient stakeholders, industry stakeholders, and collaborate to bring medical products that are safe and effective to our rare disease population. Thank you.
Thank you, Tejasvi. Lily, could you briefly introduce yourself and perhaps tie in some thoughts on trial designs and other approaches that are particularly necessary and of use for those very small, often called ultra rare disease scenarios?
Yeah, of course. Good afternoon, everyone. My name is Lily Gerard. I'm a master scientist and the technical lead of the patient-focused statistical scientist group in the CDER Office of Biostatistics. So as my group's name suggests, that my everyday work is about patient-focused drug development, and that is essentially why I joined FDA about 8 years ago to specifically support this important initiative. So I think based on my everyday work and also listening to the conversations that have happened today, we all recognize the importance of engaging patients and their caregivers early, often and early, I would say, that, it should never be an afterthought. During my work, there are many days, or I go to a meeting, or I review a submission, I question, where's the patient voice in this? Have they talked to patients? Have they talked to the caregivers? This is not reflected. These questions are now, a lot of my work involves the review of clinical outcome assessments. So those questionnaires, the patients are expected to complete, or caregivers are expected to complete for their younger children. Then I look at the questions and I question, whether patient input or caregiver input were truly incorporated. And if so, have they talked to enough of them to achieve some certain level of saturation? And, oftentimes, I want to just point out that, while we are regulators, we are also patients and caregivers. Many of us are. And, as a mom with a young child, I'm fortunate enough to not have to worry about rare disease, but there are things, that I can take off my regulator hat, put on my mom hat, and just look at questionnaires that our caregivers are expected to complete. And then from my own perspective, I cannot answer these questions. Expect that patients and their caregivers to have a full understanding on how to select these things, it's extremely hard. So I think, I agree with everything my colleague, Joshua, just mentioned, and all the other FDA colleagues have mentioned, that it is not just about talking to patients, it's about talking to them and asking the right questions. And then how to incorporate that, how to make our patients and caregivers understand that the importance of collecting information from them, how those information will be used, and most importantly, how those information will be communicated back to our patients and caregivers, so they understand that their contribution resulted in something significant. So I think oftentimes there is a gap in that I see, that the communication back to our patient communities. And so in terms of the trial design, I want to just echo what Joshua mentioned about natural history studies. Natural history studies has a very important, role in our review work, because it certainly helps us understand the disease presentation, the progression, and the appropriateness of the trial duration. But I have to, do mention that there are some challenges with just using the natural history data, right? So natural history data has to be well considered and well conducted in order to serve its intended purpose. So some of the challenges with natural history is that, what we have seen in some of the adult population is that the patients in the natural history study may have a more severe disease presentation compared to the trial population that's targeted. And then in some of the pediatric rare diseases, we found that the natural history patients tend to be older than the patients we would like to enroll in the clinical trials. So all of these will contribute to certain degree of scientific uncertainty, as we may not have sufficient natural history comparator to our treated patients. And then also, what I find frustrating from my own perspective is that natural history studies typically do not include patient caregiver perspectives, and could and should, absolutely should. So incorporate the patients and their caregivers, when you design natural history studies. And then also the natural history studies that use clinical outcome assessments often use clinician severity scales that are intended for coarse measurement of change, and may not be considered acceptable for clinical trials, because we really need to make sure the measurement, the COAs you use can be sensitive to any changes in the patient's or underlying condition. So choosing good measurement can set future clinical trials for success. Thank you.
Lily, thank you so much. A couple things here. Connie or others, can you respond to some of the more challenging aspects of incorporating COAs and natural histories? And then after that, I do want to return to something that you said about midway through, Lily, related to that bidirectional communication. And it's something that's come through a lot on the chat of trying to figure out, how to improve when possible, kind of the dialogue of making sure that patient groups receive, when possible, more insights on how information was used. That's come up a decent amount, and also how much and how one can try to work to ensure that the patient advocacy groups and patient community is representative of that community. So we'll go back to that. But Connie, John, it looks like you both come off mute. Can you talk to some of the challenges about COAs and natural histories, and maybe how to navigate some of that?
So I can start. For us, so the patient reported outcome measures, that is what we're talking about, right? In terms of is that what you mean by COA? Okay, thank you. So the trickiness of that is for us, we did not have any already out there measures that were appropriate for our population. There was nothing that could be validated in a way that could be used in a clinical trial. We just couldn't find anything. Again, the disease is not homogeneous. There's a lot to it. So we wound up having to have one made for us. We worked with the University of Rochester to create a patient-reported outcomes measure that's specific to our disease. This is expensive, and this is one of the hurdles. We were fortunate that one of the drug companies that we work with agreed to spend the money to get this made. It also is time consuming. It took 3 years from the beginning until the tool was completely completed, and it still hasn't been qualified by FDA. Not that FDA has refused, it's just that it hasn't gotten that far yet. So that's a challenge. And then the extra challenge is FDA and the FDA considering it to be fit for purpose. And so what is important and impactful to patients may not be what FDA considers to be something that can be moved by a drug and therefore be fit for purpose for a clinical trial. And I'll stop there and hand it over.
Yeah, I would agree. The natural history, at least in the case of the use of treatment of athymia and the use of thymic tissue, was really driven by the fact that early on, when there was no way of screening for these children, many of the children were dying before they had the opportunity to even be properly diagnosed. Major frame shift occurred when you have universal screening now for this underlying condition, and now you've rebooted the natural history, because now you're starting from birth and starting with interventions, and that has to be taken into context because it really speaks to the optimal timing for the initiation of treatment, and also for the long-term follow-up and long-term complications. So I think that incorporating the natural history, particularly for these pediatric diseases, is extremely important, as part of the whole clinical trial spectrum.
Kim? Thank you, John.
I just wanted to maybe part of the story that I didn't tell related to Farah's journey is the one about the clinical outcome measure development and natural history study. And briefly, I think the important learning that we've had is this is just I wish it was one and done, but it's not. It is a continually evolving process. And so we started back in 1999 with our clinicians and our patient community getting together to come up with the first clinical outcome measures, both clinician rated as well as PROs. And that was done through an NIH workshop. And those clinical outcome assessments became part of what we collected in our natural history study. And as we gathered more and more data on those assessments, there was a process where, we engaged FDA, we shared data, we talked about those outcome assessments, we got critical feedback on how to maybe modify them and continue to improve them. We learned more about them and their utility, their pros and cons through additional clinical trials. And it's, it really is this constant evolution, and the need to continue to evolve these. And when you think about new therapies that come on board, we still need new outcome assessments because there might be other symptoms related to the disease that you want to capture, or other treatments that are going to require you to assess things in a different way. And I think that's a real challenge for our organizations, because these are really long-term investments. But it is the reality. And I also think that we got advice early on from folks who at least helped manage our expectations about the fact that this was going to be an iterative process, and that we would need to be committed to that and continuing to kind of come back and refine and come back and refine over time.
So thank you for that, Jen. One of the things you're talking about, at least the way that I'm hearing, is you've had a lot of success with kind of that bidirectional communication, this iterative communication. Do you have any recommendations for others? And frankly, I'd love to bring in Lily after hearing from you and other patient groups on, well, you, John, and Connie. Any recommendations for how to maybe improve or maximize the opportunity for that bidirectional communication? It is something that me and the rest of the project team heard a decent amount about from rare disease groups, that they don't always know how things were, how their voice was used or incorporated, don't always get feedback. It was very hard, 7,000, 10,000 rare diseases, et cetera. But any advice on how FDA can improve the bidirectional communication?
Well, in our case, I think partnerships with patient organizations and partnerships with academic and clinical societies that deal with these particularly rare diseases. Most of these families are receiving their care through academic centers. So we had considerable success with partnering with the Clinical Immunology Society, and also developing the need for the long-term follow-up studies as well as the natural history studies, because in our case, the treatment is relatively young and it could definitely be modified and improved. So knowing what the outcomes are in the post-BLA period is going to be extremely important as we come up with new modalities for treatment. So it can't be one and done. It really has to be an evolutionary process. And the question is, that cost should be shared by industry, by academia, as we go forward. And I think the weight can't fall on one group.
Lily, Tejasri, any thoughts about kind of improving or maximizing bidirectional communication when possible between patient groups and FDA, and certainly, how to incorporate the reality that this is very much a sponsor-driven activity too. We can't sidestep that. What can you share? What recommendations?
Sure. So, I think that it's very important, like you said, sponsor engagement, getting patient input in their product development programs. But as FDA, we've also reached out and requested patient listening sessions. As we've had, some questions about some of our product development programs for some of these rare conditions, we have requested patient listening sessions, and we have found them very, very helpful, particularly in the context of benefit risk assessment and calculate. What is the trade-off that they are willing to accept? When we're here, we may have our own ideas, but patients are the experts in their disease, and really hearing their perspective. And so when I look at files for some of these conditions where we've had listening sessions or patient-focused drug development, I will go back to review staff and say, hey, this is what we heard. What do you think about this? So we do incorporate this. And I'd like to just sort of point out a couple, of our marketing applications. So you heard the story of Ruthymic earlier, and and from our perspective, although we didn't have any particular PFDDs or listening sessions for congenital athymia, we did receive several letters from our patient community, independent of our BLA review, that we reviewed and took careful consideration of. And then we also looked at some of the challenges that were conveyed regarding caregiver, family burdens, etc. And myself personally, as Lily mentioned, we're here, we have this job, we do this, but we're also members of this human society. We have our own challenges. I actually have a extended family member with a rare disease, and over the course of 2 years, you see them deteriorate right before your eyes. You see the huge burden that this is presenting to my family. And so, we appreciate the perspectives, and as much as our sponsors can support engagement with our patient community, and I think that, we also are looking for opportunities to engage in the context of regulatory scientific meetings between our sponsors and patients. We had one meeting where the sponsors actually brought one of the patients, and it can't tell you how impactful it was. Understanding the disease, understanding the manifestations, but then seeing a particular child who has significant functional disabilities, and then they bring the child after gene therapy, and then you see how much improvement there is. So all of these opportunities to engage are really, really helpful and actually very impactful to us. There's also, when you look at Ruthymic, the natural history data that they collected in parallel with the data in the patients was very, very helpful in our assessment of a treatment effect. We, for one of our marketing applications, we engaged a patient representative as a subject matter expert to provide feedback and input on a particular endpoint, particular novel endpoint. So, where we are able to, we are seeking opportunities, and we will continue to collaborate. Thank you.
Connie, thank you. Connie, I think you were next,
Yeah.
and then I've got one more biggie that I would like to introduce before wrapping the session. Connie,
Okay.
please.
It's very simple. Hold your meetings in the Washington, D.C. area, and that way you can invite FDA people, make sure they get scheduled enough ahead of time that they can clear their schedule for the day, have them come, be a speaker, meet you all, meet your researchers. I mean, seriously, they need to get to know you, and maybe they don't always have the opportunity to bring you into their house, but you can invite them into yours and make sure it's at their back door.
Okay. Helpful. Thank you. All right. One last big one that constantly comes up in the work that we do on topics far removed from rare diseases, say innovative trials, all sorts of different things. Come up a lot in the chat too. Can FDA folks talk about efforts to try to consistently integrate patient engagement? Listen, there's what, 1,100, 1,200 reviewers in Office of New Drugs, right? And across therapeutic areas and even within them, but there's just across therapeutic areas, you think about the neuro space where you might have a lot more exposure to rare diseases, to patient engagement type stuff. Anything FDA can talk to the patient community about efforts to kind of, as much as possible, improve consistency for that patient engagement across review divisions and centers. Anything you can share there, and any closing words from Connie, Jen, or John in that regard would be helpful before we take a very brief break before our final session of the day.
Yeah, this is Lily again. So thank you for that question. I will, be frank and admit that, maintaining consistency within a large agency like ours is difficult. But we are trying our best, very best, to make sure that, messages, recommendations, considerations are communicated in a consistent manner. So I'll take my own team as an example, because it's my team's special function. We don't work directly with just one therapeutic area in, the Office of New Drugs. we work with all therapeutic areas. So we have a chance to touch each of the review divisions and remind them that you need to incorporate, make sure that in your considerations that you ask sponsors to do the amount of work that's needed to incorporate patient voice, to get their understanding. Because, in many of these rare disease areas, I cannot say that, we are all experts, certainly I'm not as a statistician, so I rely on my clinical colleagues to help me fill in the gap on what the disease is like. But most importantly, I want to hear from our patients what their experience is like. So in many of the areas, yes, we do have patient-focused drug development meetings, whether it's internally led or externally led. And now we are seeing more and more of these patient listening sessions, which has been extremely helpful in my case. And then, there are also the CPAN meetings and different consortiums that, I think patient engagement can be part of, and that FDA should be aware of these engagements and then seek the opportunity to get information. So as I mentioned, that I lead a team of biostatisticians and psychometricians. They all want to do fancy stuff. I want to do the fancy statistics, all the psychometrics work to make sure that the tool we have is valid, and reliable. I tell them, before you start to do your real work, go attend these meetings whenever possible. First, listen to our patients, hear them first. You're not the expert in all of these. You may be experts in your statistic methods, but you're not an expert in these disease areas. You have to understand what we need to target on, because if we start off on the wrong path, not focusing on what patients consider to be important and that can be affected by the drug, then in the end, we're only going to get data that's uninterpretable, no matter what fancy statistics you use. So I think it's just really important that right from the beginning that, we, talk to our reviewers, talk to our divisions who may have less experience in these areas, get them up to speed. We have lots of internal presentations across the clinical divisions, across statistics divisions, to help them understand why it's important that we have this work, we have this initiative. So I think, again, we're working, our best to make sure consistency is exercised across the divisions and also, across centers. But, we just want our patient communities and our other stakeholders to understand that we are here to collaborate and to support, and we listen to all the things, and we will do our best. Thank you.
Thank you.
Can I add some, just very briefly? I echo
Yes.
everything Lily said, but I'd like to see that even for these rare diseases, there may be drug products in development as well as our biologic cell and gene therapy. So we heavily communicate with CDER on these. We don't operate in silos. We discuss endpoints, we discuss study designs, and so we really do try to aim for consistency. And we actually have regularly scheduled meetings now that where we're trying to, in our effort for consistency with our rare disease group in CDER as well. And I think the one comment, I think Connie, you had made regarding, have meetings in FDA's backyard, please invite them. I think we are more than willing to come and attend these meetings, learn from our patient community, and then incorporate what we hear from our patients.
Thank you, Tejashri. We're up on time for this session. Connie, Jen, John, Lily, Tejashri, thank you so much. This isn't an hour that you devote. This is every day, every week, every year. We know that. We're grateful to all. We're in it with you. And let's take a break. We're back in 8 minutes. We will bring everybody back. We've got a lot of critical voices that have been involved in all this stuff for decades. I'm going to try and tie as much of this together, building on next steps to follow.
Hi, everyone. Welcome back. I know that was a short break, but really appreciate you all joining back on and sticking around with us for this final session of what's been a pretty packed day, half day anyway. So my name is Tori Gem, and I'm a research associate at Duke Margolis. I'm honored to be serving as the moderator for our last session of the day, where we'll hear from really a set of wonderful panelists who will share some reflections on what effective approaches for patient community engagement have looked like to date, some of the significant remaining challenges, as well as thoughts and recommendations on really how to have more effective and efficient incorporation of patient experience and input in the future to really best support rare disease drug development. So with that, I would like to welcome our panelists for this session on screen at this time, and I'll go ahead and briefly introduce folks before we do some brief remarks from those who are joining now. So first, joining us for this session, we have Karen Holzer, who's the Director of Policy and Regulatory Affairs of the National Organization for Rare Disorders. We have Colin Hovinga joining us, who is Vice President of Rare and Orphan Disease Programs at the Critical Path Institute. We also have Jennifer Panagoulias, Head of Regulatory and Policy for the Foundation of at Angelman Syndrome Therapeutics, and also co-director of the Angelman Syndrome Biomarkers and Outcome Measures Consortium. We also have Saira Sultan, Lead Policy Consultant at the Haystack Project, and then Bobby Weissman, who is a patient advocate and a person with lived experience. And then lastly, we'll have Carrie Jo Lee, Associate Director for Rare Diseases in the Division of Rare Diseases and Medical Devices, Office of New Drugs at FDA Center for Drug Evaluation and Research. So before we turn to the discussion, start with some questions and really try and cover as much audience Q&A and some of the themes from the audience Q&A as possible in this session. I do want to give folks some time for really brief opening remarks. So first we'll turn to Karen, if you'd like to just share some brief thoughts to get us started.
Fantastic. Thank you so much for having not be part of this important conversation. What a great conversation already today. Patient-focused drug development, patient experience data has been really central to NORD since we were founded really as an outswell of patient engagement 40 years ago. I think reflecting on the conversation we had today already and what we're hearing day in, day out from our community, I think I want to highlight three common challenges. The first one is around common language. I think there are lots of different terms that we talked about today. Patient-focused drug development within the terms of a PFDD meeting compared to all the different ways in which patient engagement and patient-focused drug development can take shape. And I think it's really important to be clear which of these we're talking about, because it's very easy to talk past each other. If you think about patient input in a clinical trial versus a PFDD meeting, both are very same thing. Then also we heard a lot already about realistic expectations. And I think that is tremendously important. Just making sure that everybody's on the same page. We heard a lot about where, for instance, PFDD meetings can and cannot be meaningful and how important timing is. And so really being clear about what that patient engagement looks like, what the goal is. And for instance, that a voice of the patient report can be really important in forming trial design, but maybe is not a good substitute for a primary or secondary endpoint in a pivotal trial. And so having that expectation, having that clarity of purpose from the beginning. Talking about fit for purpose, and that includes representativeness, understanding that everybody in our community feels the same way based on their lived experience, based on where they are in the disease journey, and really making sure that we purposefully include all of the voices in the community. And then there was a lot of focus on transparency and accountability, which I think is really important to trust, which ultimately is foundational to patient experience and patient-focused drug development. Then lastly, I loved the conversation earlier today about what else can be done with regard to patient group interactions with FDA beyond the PFDD and patient listening session. We oftentimes hear from patient communities that have had a really great PFDD meeting, but they want to continue to engage with FDA. And so I think talking more about the other ways in which patients can meaningfully be engaged in patient-focused drug development is really helpful. With that, I'll hand it back to you, Tori.
Great. Thanks so much, Karen. Next, I have Colin, if you want to give some brief remarks.
Yes, sure. Thank you for having me and representing Critical Path Institute. And I guess I was told that this should also reflect my past lives. So I'll try to comment to the different points that I've heard, but also thoughts of like where we should go or where I see our future. First of all, I felt really old listening to some of the speakers. I don't normally see myself as old, but this presentation's made me feel old because I can remember a time in which, as a resident, asking different parties about funding a project and being told we don't care because this doesn't make us any money. And I go from now into a period of time where there's really active investment in drug development in rare diseases where it wasn't as much of a precedent. So in my lifespan of where I'm at, I know I don't look that old, but I actually seem to be, there's been a dramatic change. And I think that's been a good thing. And I see incrementally this improving over time. And I see a lot of success stories, in meetings that I've been at for rare diseases that have had successful discussions. I think there's also been a push for more holistic indications. What I mean for that, concepts are really relevant to persons with conditions that we wouldn't normally think of, so not necessarily direct targets of a mainstream that are easily captured. So it's really set up a more, I guess what I would say, more patient-centric view of drug development. The other element as being, sort of a trialist in a person operation trials, then I put that hat on. The other element I see a lot of encouragement and a lot of news is that there's been increased interest in decentralization of clinical trials and tools and measures and infrastructure that can really foster that. Because one of the things I really hated as a clinician and as a researcher was I have to watch families travel miles, with their durable medical equipment into the hospital, across the clinic, take them to different departments, just the train wreck of that, really trying to build infrastructure and trial methodologies that would be sensitive and also get feedback, from individuals that are impacted with the disease and their families. I think the other part of this is there's been a great increase in empowerment. I mean, one of the things I know that we do, and I know that NOR does, and there are other organizations that really work with the community to try to address, some of their challenges with their disease, data collection, creating natural history efforts, really, creating that infrastructure so that they can begin to understand their disease in ways that they may not have had. And really helping them, connecting them with the expertise that are critical for that understanding. And I think one of the last things I'll comment on is that I see there's a lot more connectivity. Like as an academician, I didn't really understand that sometimes my clinical research that I was doing or my lab research may not have a direct translation into drug development. And I didn't understand that because it wasn't speaking the language that my other parties needed to understand. And so one of the things I see increasingly happening and over time has been this interesting dialogue between like international community, nonprofits, federal funding agencies like NIH, and FDA, all to really tack things very strategically so that we're using the science in ways that we normally didn't. So industry can build upon these things for new treatment and de-risk rare diseases in specific ways that they couldn't before. But I will stop there with my comments and pass it back to you.
Great. Thanks so much for those comments, Colin. We'll turn to Jennifer next.
Thank you so much. I'm so pleased to be here today and representing the Foundation for Angelman Syndrome Therapeutics, or FAST. And I'm really, I want to thank the organizers for inviting me to reflect on where we've been. Angelman syndrome is a rare monogenetic neurodevelopmental disorder for which there are no treatments. And so at the Foundation, we have a number of work streams to really help inform clinical trials. And I came to advocacy from industry because my family was touched by Angelman syndrome. And when I think back 20 years ago, because I'm also old, and how we design trials in the rare disease space, we never asked patients or caregivers what they thought about trial design, which is a terrible oversight and a terrible way to design a trial that you want to be enrolled. So what we've tried to do through the efforts in the FDD space and through the guidance series that FDA has issued and the work that they've done to really make sure this key stakeholder is incorporated as part of the process is to collect this caregiver input from our community and make sure it informs clinical trials. And so we've spent a lot of time, and I think tremendous progress has been made in this area to bring that voice to the table. We ask for them to provide us information through surveys and other means on symptoms that are burdensome, what the disease burden is, what do they want to see change, what does meaningful change look like to them, and importantly about trial burden. And I was glad to hear a lot about that today, because we really need to understand in designing trials what is too much. And so we've collected information through caregiver advisory boards. We facilitate and participate in these advisory boards specific to trial design on specific protocols. And we get a lot of good information. And things that have come to light are about what is too much. And I think about inputs from families where they say, well, we're going to be part of a trial, or I was part of a trial, and it has a complicated administration procedure. And my child had to arrive at the clinic and have a physical exam and then perform outcome measures. They had to go through a series of performance-based outcome measures. There were clinician-directed interviews. There were questionnaires, there were labs and imaging studies, and they needed anesthesia for the administration procedure, so they couldn't eat. And we haven't even had the drug administered by this point. So we're 12 hours into it, and that's not feasible for a family to undertake. We're talking about one visit that really has to take place over 2 days, and maybe the family lives 3 hours from the site and they have to stay overnight. And so this impacts trial design. We don't want to see trials not be enrolled, and we don't want to see a significant amount of dropout. So it's really important that we collect these inputs and they factor into regulatory decision making. And so where I hope we're going with all this is to strategic partnerships between FDA and advocates to make sure that we're collecting this patient experience data on outcome measures that are important to them and trial design, that it gets really factored into that process and it really does influence regulatory decision making. What's really important is that our caregivers and our patients sacrifice time and effort, and they fundraise for us. When we undertake these efforts, this money comes from our community through bake sales and lemonade stands and golf tournaments and what have you. And so it's really important that we spend that money judiciously and that we know that it's going somewhere. So these partnerships are important to make sure what we're collecting is meaningful and really does have influence on the process. And I'll end with this. I really appreciated the comments and the presentation relative to Bayesian modeling, because we'd really like to see this patient data and this trial data really evergreen the development process to move from one trial to another to influence study design and to decrease sample size.
Great. Thanks so much, Jennifer. Syra, if you could go next with some brief remarks.
Thanks for having me. I second what everyone has already said on this panel, so I'll take a little bit of time just to say that Haystack Project is a nonprofit. There's about 140 plus rare and mostly all ultra rare patient groups in the organization. It's been around for about 9 years now. We hope that we'll provide you some meaningful input today, and in future engagements, we really thank Duke Margolis for including us. And we hope FDA staff walks away from this meeting seeing us as a resource and uses us beyond the usual suspects that you go to so often. Not that they aren't wonderful, they are. But new groups don't crop up if there's nothing new to add. And so we are here and have been here 9 years, and hopefully you guys will continue to engage with us. Given that I only have a couple of minutes and this meeting is really about increasing and improving the engagement patient groups have, I want to share some very tangible examples of where we have been recommending change. So in the area of advisory committees, FDA just put out a call for nominations for genetic metabolic diseases. And we thought that was wonderful, and a recognition that perhaps some new experts were needed at the FDA. But I want to remind the agency that without changing your conflict of interest rules in how experts engage with you, you won't be able to use the experts that you're compiling and getting nominations for, because almost always you'll have 2 or 3 experts in a rare disease, all of whom are engaged in research. And so there's got to be a way to involve and include them with disclosures of what their conflicts are. After all, they're not speaking to sort of naive people. They're speaking to fellow committee members who are very knowledgeable in their own right and know how to discern important information from not and how to weigh that conflict of interest. Also to include treating physicians, also to not refer patients or treating clinicians just to the quote, sort of open mic time you have. I think that they have so much more to contribute than just what they can in the 2 or 3 minutes they get for open mic time. So really, really important. There was a recent Pink Sheet article on how EMA is doing this work, really very proactive with reviewers reaching out ahead of application reviews, involving patients, and now they'll start involving HCPs. They look back over a month or so as to what applications are coming and try to engage proactively outreach to patient groups in advance. And I thought that article was really worth a review. On PFDDs, I heard Robin say that FDA can't say how reviewers use PFDD data, but what you can do at FDA is ask questions, ask clarifications, engage the people who have spent an inordinate amount of time and money and resources putting those PFDDs on. People aren't actually engaging in those meetings when they're happening. So we don't expect FDA to be experts in every rare disease, of course, but we couldn't have possibly told you everything there was to know about the disease and you had no follow-up questions or clarifications at all. So how can that engagement happen? I think it happens well in listening sessions, which by the way, cost the community far less time, money, and resources. And I think they're a far better tool than what we're doing now with PFDDs. Also, you don't have to let go or forego any confidentiality with sponsors if you're simply asking questions of the very people presenting about their own experience. You can do that without breaking confidentiality. And by the way, patient groups know how to sign NDAs too, and they do it all the time. So use them the way you need to use them and go back to them throughout and help them understand if they want to affect endpoints and they want to affect clinical trial design, tell them when to do their PFDD. But if they want to affect a reviewer's understanding as they're getting ready to review an application, that's a much later time in the process, and they can hold their PFDD much later and engage much later in the process. I think that'll reduce some of the frustration from the patient groups who are conducting these PFDDs. REMS. We have REMS provisions that tell patients they have to do things or doctors they have to do things, but we don't always reach out to the patients and the doctors to ask, can you do it? Will you do it? It's not okay to assume that patients just simply won't, and therefore benefit risk will go cut against them, whereas it might have gone the other way. And then finally, I just want to quickly say, so much great information. I really appreciated what Lily said and Tajajri said. I'm sorry if I'm not pronouncing that right. I so appreciate where you're coming from. We know FDA does many things well. We know you guys have an impossible task ahead of you. When we see you guys say, look, we're parents too, and we appreciate how important this is, we get that, and we're not ever questioning that. What we would say, though, is that we are told as patient groups all the time, we cannot legislate or regulate on the basis of one example or one anecdote. We asked that you all take some time to figure out how you are consistently using the resources patient groups are giving you. Who's using it, which reviewers are doing it, how they're using it, document that so that your leadership can see where it's going well and where it's not going well, so they know which resources to provide you. And are you using the educational resources provided you? We know you have them, but we also know you're busy. Right now, our patients feel like it's a lottery which division they're assigned to and as to whether they'll get an application through the process or not, or even get it accepted for review. And that shouldn't have to happen. Patients really need to know that whatever review division they end up in, that they will be able to get a fair hearing. They want a reviewer who will take the time to talk to them, who will understand how accelerated approval works, who understands whether they can or cannot meet those hurdles, who can understand that we can't just say we need another trial when there aren't any more patients left to do another trial. These are, of course, there's wonderful reviewers at the FDA, but consistency across division and having the leadership know which reviewers need extra help and don't I think is really critical for you all to know and also for our community to know, because then we can all legislate and regulate with data. And right now we don't have processes in place at the FDA for these sorts of things. Those are a few of the tangible solutions and improvements we'd like to see. I could talk about many more, but I'll stop there.
Great. Thank you so much, Syra. Appreciate you covering a lot of, specific recommendations, and I'm sure we'll return to more thoughts there as well as we progress in the conversation. All right. So last but not least, I'm a man of many hats, but Bobby, would love to turn to you as your experience as a patient and as a patient advocate for some brief remarks.
Victoria, good afternoon first, everyone. Let me be clear of my name, because it's Robert Lee Wiseman Jr. And for this specific meeting, it's Minister Robert Lee Wiseman Jr. And that I'm restating it. I'm coming from unsiloed kaleidoscopes, fragmentation, and non. As a person with 4 chronic disorders, diseases, IC9, what all that stuff is, one part is not greater than the other. My clinical teams, my family, they got all different parts, but coming together to be in a trial, both on the pre, post, during, and all of its wonderfulness, to say the magnitude of the voice of a silo, unsiloing those voices to where the kaleidoscope comes together. Because I seem to recall the title is where we have been, where we are going. The we is not really me, it's a we. That's a collective, that's a collaboration, that's a solidarity, that's a unity that says we can have conversation in the midst of discourse. Because I think, what's her name? Dr. Nicole Verdum. I probably got your first part wrong. Said it, voice. There are voices in all of this. One voice cannot speak without the other voices. That is key to all this. So I've gathered some time back on up. And for Baptist Kojic, I'm doing good right now. And just finished something else. Yeah, I said it. I'm very clear. Because the passion that I have and continues is because what's been done for me by others who did not agree, but found a way to come together. That's it for right now.
Great. Thank you so much, Bobby. Really appreciate highlighting the, path moving forward together as sort of the path forward, and collaboration is key. So, all right, with that, I know we've got a lot of ground to cover in the approximately 40 minutes we have remaining. We want to work in as many sort of questions from the audience and sort of themes that we've heard, both those questions we got before the workshop as well as ones we've gotten in the Q&A here. Before we get to some of those, I do want to start with an initial question. I know you've all highlighted and touched upon or sort of provided, some deep thinking on how impactful sort of patient input has been to date, as well as kind of the reflections on where we've been and how the space has evolved. So would love to hear, if anyone would like to jump in with additional thoughts about how impactful patient input has been to date in the drug development and review process, and sort of some remaining challenges that we might want to discuss.
I will go that the wordsmithing of it in the drug development process, I think, and know that starts at the local level. What I mean by the local level, in the home first, then to the doctor clinical office, then it builds its way up, because all those there's the qualitative, quantitative, and anecdotal, the person side of it. And I think where there's the schism of it is how that's translated on both sides, from patient, provider, researcher, for the maker of it all, of bringing the richness together where the document becomes living of what is it that needs to be developed and the why, where we shift away necessarily from an ROI of a fiscal value to the ROI of humanity, the ROI of how this will increase quality of life, of how this will build resilience, of how this will build a new way of communication, and how it shows its adaptability. So for me, in my experience, both with a person with a bleeding disorder, my HIV side, my hepatitis C side, my pulmonary arterial hypertension side, all of those different spaces, and being a person of color and a youthful 52 and a half come December 25th, that's my experience of some of the challenges. Because I am young still.
Great,
And
thanks.
maybe
so
I'll
much,
build
Bobby.
on a little bit of what Bobby has said as well, that I think it's very difficult for us to say how far we've come in patient engagement, because while there are new platforms like PFDDs that did not exist 10 years ago or whatever it was, 15 years ago, we aren't using them effectively as far as we can tell as patients, or if we are occasionally using them effectively, because there's certainly good examples of where PFDDs have worked and worked well. But unfortunately, there's far more examples of where they haven't. The other thing I'd say about how far we've come is if it has gone well, we don't know as a patient community when it's gone well. We don't know how reviewers are using this data. We don't know when they're using it. We don't know how they're sharing it with other review divisions. And so again, we have to come back to collecting data. We get accused, as I said, so often of patient groups not working from data, but in this case, we asked during the last PDUFA cycle that their FDA provide us data on these different things. And they have a fair point that if you provide too much data, it's very easy to unblind which particular application or disease state or company might have been involved in a particular application. But there are thoughtful ways to not just sort of leave it off and say we can't do it, but maybe just take a scalpel and remove 1 or 2 data points that would make that confidentiality stay intact, and then share the rest with us so that we can all decide where it's working and where it's not. But today, I don't think we can say.
If I may just give a slightly different perspective, and I think part of it goes back to how we talk about patient-focused drug development. I think we do, what the workshop today really reinforced for me is that patient-focused drug development really goes throughout the whole drug development process, all the way from discovery or through product life cycle. And I do think we have come a very long way there. I mean, the fact that we are here today having this workshop, I think 10, 15 years ago, that would definitely not have happened. I think we have a lot of examples where patient input has meaningfully impacted clinical trials, where patient-focused drug development meetings have had tremendous impact on the trajectory of the drug development process, where patient-reported outcomes have made it into labeling and made significant impacts for patients and the drug development process. So I think we have come a long way. I completely agree with you, Sarah, that we're not there yet. I think, to Bobby's point, making sure that we fully represent the totality of the patient voice, the experience, patients as complex members that have multiple diseases, that live in communities, and that have a lot that and multiple silos, as you so aptly said, Bobby. That is, I think there's a tremendous potential to further the drug development process and the voice of the patients in drug development. But I definitely think that we've come a very long way, and I really appreciate FDA's long-standing work on patient-focused drug development.
Yeah, Karen and Sarah, thank you so much for those comments, and certainly appreciate that there are a variety of sort of markers that demonstrate and indicate progress, but appreciate that more sort of communication and information really could be valuable for demonstrating how we can, you know, further this work moving forwards. Really appreciate that. Colin, it looked like you want to jump in.
Yeah. I mean, I think one of the things I keep hearing in these discussions is a lot of FDA, FDA barrier. I can tell you, on being on different elements of drug development throughout my life, it's a broader ecosystem that we all need to consider as we're looking towards this. Because, I mean, like, for example, I mean, it isn't. I worked on a working group at one point and not too far ago that was talking about patient engagement in designing protocols and reviews, and we work with sponsors and whatnot. And there was a large diversity on whether or not you even were required to have any input from more than one consultant, even externally. And these are so, let alone a patient, providing any insight to a protocol. So, I mean, and then we're focusing just on the US. I mean, it's a much broader ecosystem that we all need to consider as we're looking through this, because unless we're all, and it's not rowing in tandem, that whatever is the most conservative reaction in getting us to move forward is the one that's going to cause us to move more slowly, right? So, I think that educating and encouraging sponsors to, as well as other agencies to really engage with the patient community. It's essential. And I think the last part of this is that when you look at this from when you look at this at a static point in time, it's hard to judge where you're at. I mean, as I always say, I'll echo Karen again, is like, if you've been doing this for a while, we've actually come a long way. It's not saying we're perfect, but there has been marketed increases and successes in this space. And I think it's through these collaborations that we're only going to do better as we in the next coming years.
Great. Thank you so
Let
much,
me just
Colin, for
clarify
those.
a quick point, Victoria, because I want to agree with my co-panelists here. First of all, I might be aging myself, but I've been doing this work for 30 years. Literally started with the very first PDUFA reauthorization back in the 90s. So I will say that we have watched the evolution, and we are in no way saying that that FDA has not made enormous strides and continues to do amazing work today. We want to see safe and effective drugs on the market as our patients in ultra rare diseases, just as much as FDA does. We want confirmatory trials just as much as FDA does. We don't want access to treatments that are not going to work for our patients. There's too much risk for our children, as you've heard Connie and others say earlier. So I think I just want to clarify the point that I don't think any of us doubt what great work FDA does, but a session on how we improve and increase engagement at the FDA, not overseas, but here, using perhaps overseas examples as I did, I think that's the goal. So I think we're all on the same page there. Just want to clarify that.
Thanks so much,
Yeah.
Syra. Carrie Jo, go ahead.
Yeah, sure. I wanted to take a moment and just, I think that listening to the question and the answers, I think there's a lot of shared goals here. I think we've made incredible strides. I mean, not just in terms of drug development, but in medical care, in moving towards a more patient-centric focus for how we approach, working with patients and collectively trying to treat diseases. I will say that I also think that it's incredibly important to take a bit of a step back. And what we've heard even today, which has been really informative, is how the patient voice needs to be heard throughout the drug development process, which starts much earlier than the FDA getting involved. It's in the academic universities, in the clinical trials, that start with sponsors. And so having that patient voice available, whether it's through the patient-focused drug development reports or the patient listening sessions that have come to the FDA, or whether it's through engagement through workshops in the pre-competitive space or consortia in the pre-competitive space, it's important that everyone hears the patient voice and incorporates that into drug development from the beginning.
Yeah, thank you so much, Carrie Jo, and really appreciate sharing that. And I think that we've heard that sort of throughout the day, and then completely appreciate and recognize, Syra. I think your comments are heard loud and clear about some of your specific recommendations and your focus on one partner, but acknowledging that it's a larger there are a number of other sort of players in the space that are critical to engage. And I know that you're, sharing some specific recommendations. So I do think it would be helpful to sort of turn to other sort of thoughts on recommendations for thinking about, what we can do engaging with all of the stakeholders, including regulators, but also thinking about, sharing learnings across patient organizations to really understand how to do patient engagement, how they can, sort of engage in a process in a more efficient and effective manner, how other stakeholders play a critical role, making sure that they're understanding and engaging patients at the right time, including researchers, industry, and regulators. So wanted to take a moment and see if I could turn to some other folks. I think, Jennifer, it's been a minute since you've got a chance to jump in, so maybe turning to you first, and then turn to the others as well.
Yeah, thank you. I think consortia that Dr. Lee just mentioned is a great way to ensure that we have continued engagement over the life cycle. And it's really important because that does bring together the other stakeholders in the community. We obviously run a consortium at FAST that's pre-competitive and includes industry and academics and other advocates. And at times it does include FDA, and we've reached out to the European Medicines Agency as well, because we do want to have a global perspective. We don't want to run a trial for every country rather. So we want to see a harmonized development program for certain promising, therapeutics, that we have one trial that can serve the needs of all the global regulatory agencies, because we have a rare disease community. So we really need to work together to make sure we have the right outcome measures and the right inputs and the right natural history data to achieve that. So that's something that we really support. And I think along those lines, that's why I mentioned partnerships, because it's really important for us as that community to make sure that we do have regulatory support really globally at the outset of what we're doing, so that what we're spending time on in terms of looking at outcome measures and biomarkers is really fruitful and will really help ensure that we have the appropriate measures for use in clinical trials. And so getting that engagement at times is hard, because if we went through, let's say, for example, the endpoints qualification program, that might take us years to achieve qualification of an endpoint, where instead we can come together as a community pre-competitively and agree on what is really fit for purpose early on and make sure we take that forward. So I think that's a great way to ensure we have engagement.
Great. Thank you so much, Jennifer. And then Bobby, I know, again, you have lots of lived experience from many sort of hats. So I wanted to see if you want to share sort of any reflections on, from your experience, what maybe are good recommendations.
I'm trying to be good, but I'm going to just go there.
Sure.
And expectation coupled with education, anticipation versus angst, and communication and cooperation. The reason why I phrase it that way for engagement is that me being 52 and have come through a clinical trial, I know what that entailed for me. Yet my nephew, who's 21, may not understand what is that process to be in a clinical trial. Because we're not sitting at the house, dude, I was in this trial, but that's not the norm of the day-to-day of living life. And so the expectation and education, what does it look like? That is is someone's engagement going to be writing a letter? Is someone's engagement going to be collecting information orally? That if someone is, on the tape recorder, so that it's in a collective spot, so that when something is developed, there's an archive. That the cooperation of communication, coming to the table to agree to disagree and agree, and an understanding of what an AAV vector is compared to a urine cup. Yeah, that's an example of being in a trial, and to where it makes sense to the person saying it and hearing it, and the anticipation and the angst that go together of what could it be versus what it's not across the board, and the recognition of differences in geographic locations that fuel one's expectation, one's communication, and anticipation of what could be. And fundamentally bringing it down to choice of everyone involved to be willing to have the conversations and understand experiences may shift over time. And that's okay. And to be open enough to say, okay, what is engagement in the process? The process may need to be redefined based on current context. It does not negate, yet it adds to.
Great. Thank you so much, Bobby. And I know that, you certainly have shared other reflections as well on, I think the need to make sure that it's sort of a continuing conversation with patients as they go through the experience of participating in trials, and how their sort of needs may shift as well, and being responsive to that.
And so not just the patients, it's the patient's family or family of origin choice and the clinical team, because it's not just that medical file. It's all the parts where it overlaps into work, school, all those things, one's physical fit, all of that. And to where also the clinical teams are involved, boards of directors, all of that. It's not, in my opinion, a one voice conversation.
Also helping the patients understand where they're engaging and what its purpose is in that moment, to what Bobby just said, right? If we know that they're engaging because it's time to design the trial so that it is working around work and school and the things that Tom Miller talked about this morning, versus if they think that they're engaging at a point where they're trying to educate a reviewer with an application in front of them. I think you could do a lot in just separating out what the engagement, what purpose it's serving at the time. not just for FDA, but I think also just for managing patient expectations of what that's about.
Yeah, thanks so much, Syra. Oh, go ahead, Karen.
And if I may just add, education of the sponsors as well. Colin, I really agree with your point. I know that many of our patient communities have very small hamstring patient advocacy groups that have no staff or very little staff. And so setting expectations from the drug sponsor side of what is the infrastructure, what are the patient groups that are available in the space, how can you nurture them so that they can be partners over time? And I think we've heard some wonderful examples today about patient communities and patient advocacy groups kind of growing with the with the sponsors, building long-term trusting relationships, which are so central to the drug development process. But I think having some of that education that in many of our rare disease spaces that doesn't necessarily exist at the beginning, I think is really important to make sure that we start off on the right foot.
And I would agree with that. I think there's 2 points. One, the consortium model is great. CPATH is involved in a lot of consortia. It brings together, industry, regulators, different nonprofits, personal lived experience. And it's a nice, not only you're developing solutions and creating drug development tools, but it also is a great networking and lessons learned opportunity because we all see the world in our own little lens, right? So having those conversations and understanding how to address unmet need in a very strategic, stepwise fashion is very, very important. The other thing, I mean, this comes back a little bit from my clinical days and from also working with nonprofits, is that we also use the buddy system, and there's no other groups that can better understand you probably than a more mature patient advocacy group, because they know how the machinery moves and how difficult it is to start and all the questions and all those things. Having someone that can speak to you to that is, I think, it really essential. And so, there are many of the groups on this call that have just done an amazing job of working, into the ecosystem and involving. However, there are many, especially in ultra rare, that don't have that framework. I've talked to ultra rare diseases that don't even know where to start. I mean, and it's not the fault, is that they're limited budget, expertise, might be a parent, and really needing that sort of advice and maybe even emotional support at times, because it's not an easy path, right?
Yeah, thank you so much, Colin. And I think actually that leads into, we got an audience question, really, I think highlighting how can the recommendations sort of shared throughout the conversations today really be applied to the smaller ultra rare disease populations. So really acknowledging, the sort of different spaces where folks are at. And I'd actually love to turn to Syra first on that. I know a number of your member organizations are representing much smaller patient populations. So if you have any reflections on sort of the range of recommendations, sort of engagement throughout the process, and sort of considerations for applying that to smaller patient populations.
Yeah, we spend a lot of time at Haystack with those individual patient groups, often started by parents because they were handed a diagnosis for a child and don't know any other families with that same disease, and really drop everything. And I often say they have the same 24 hours in their day that I have in mine. And yet it's always remarkable to me how much they're achieving. They're not only helping their child, they're holding down a job, they're starting a nonprofit, they're finding other patients like themselves, they're finding researchers, then they're finding companies like you heard earlier and convincing the companies to participate in drug development, and then helping put them through finding the patients for the clinical trials. We cannot underestimate these patients and these patient organizations. They worked so hard through the last PDUFA negotiation, literally sitting with us one by one, talking about every single engagement they'd had with FDA, and understanding how over time it sort of became a joke with all of us to say, they would say, I don't know how to work on a law. I don't know how to work on a bill or legislation. And I would say to them, it's literally like you're sitting at your Thanksgiving table and you're talking about how your day went, and you want to say, God, there ought to be a law. That shouldn't have happened to me today at the grocery store or at this or at that. It's really no different, right? Put yourself in their shoes and help them make it easy for them. And then all of a sudden they can say, yeah, I should have gotten and asked a question at a PFDD meeting. Oh yeah, I should have my expert who's a rare disease doctor be able to not be conflicted out of every conversation and just have a general geneticist represent an expertise on a disease no one's heard of. Oh yeah, there ought to be a lot, they can think through these things. They can take their experience and put it into words that lead to tangible change for them. You know, we're all sitting around this table, many of us, with policy constructs that have governed our days for 30 plus years. They don't have that kind of time. Their child's getting worse every day, and they want to see some tangible change, whether that's among the researchers, and I mean this throughout the ecosystem. I'm focused on FDA because this meeting is supposed to be focused on FDA, but of course the change in ecosystem is really important throughout, that all of you have said as well, researchers, drug companies, et cetera. But I think we should not underestimate what these groups get done in a day compared to what I get done in a day, and many of us get done in a day. So that's what I would say about them.
Great, thanks so much, Siren. I think if we could, I think
Fine.
that leads nicely to just getting some reflections from others on really what can sort of stakeholders do to be good stewards of patients and caregivers' time and resources. So Karen, I'm not sure if your comments were going to sort of lead us in that direction.
Yeah, I just wanted to give a slightly different flavor to what Sarah said. About half of our 330 disease-specific rare disease members are really small, no or one full-time staff member. So we deal a lot with the startup patient advocacy groups. I think absolutely agree. It's amazing to see what parents and caregivers are doing, standing up. And I think we heard some of these stories today, standing up amazing organizations that are really going out to change the world. I think my recommendation would be first to start somewhere. I know it can be very overwhelming to think about setting up a patient-focused drug development meeting or patient listening meeting. But I think what we heard loud and clear today is there are lots of resources out there. There's FDA staff there to help. We at NORD are regularly working with patient groups to help them through the process. Similarly, to what Colin said, Facebook and peer-to-peer mentoring has been tremendously helpful for many in our community. Working with drug sponsors on how to engage, again, there's a lot of opportunity to learn from peers. But I think my first recommendation would be just to start somewhere, not to get paralyzed.
I think they need to know that when they make that effort with NORD's help, and I know NORD has helped a lot of groups put on PFDD meetings, I think that they walk away feeling like it didn't achieve what they hoped to achieve. So I think managing expectations is really important. I do think they can do the PFDDs, they do them. But I'm not sure that what they're sharing with each other, at least on the calls that I'm participating on, the answer is, tell us how you used it, and then we'll continue to do it, right? That's the distinction. I'm not saying they shouldn't do it, or it's not a good resource or a good platform. I'm just saying they need to know that somebody heard it and used it and how, so that more of them keep doing it.
Yeah, thanks so much, Syra. I think that's a theme that's come up certainly multiple times, and really appreciate that the communication back, like the ongoing communication, and not just having that PFDD as like a single time point is certainly, I think, an important recommendation. I do want to shift a little bit. We do have an audience question that I think would be helpful to sort of turn to Bobby on, because Bobby, I know this is something you're interested in potentially talking about, is really understanding how can efforts to engage patients be improved to reach the whole population, including those who may face barriers at accessing trials or involvement in drug development. So how can we really ensure we're engaging the whole population so we understand and can meet community and sort of improve a diverse set of participants to get us started off on the right foot on that sort of journey?
I strongly believe and know to have a diverse population in a trial requires something very simple: communication. Communication that is not dictated by a Mr., Miss, doctor, nurse, all that. Person to person. Let's talk. That yes, we can have a scheduled call and listening, this, that, the other, but a sit down let's get to know each other person to person and to hear their expectation. There might be a researcher who has a thought to research on and has been working on it and has been very focused in lab, yet not seeing the connection to an individual, to a person in his or her or their community. There might be a family that's been struggling to get access to something for improvement of life, and they've had roadblocks, insurance kerfuffles, just problems, and they have a feeling or an angst to a researcher. But having the people sit down and speak in trueness, person to person, because at the end of the day, whatever is developed or not, created or not, there's a human being behind it. And I personally feel that will help spur engagement. Engagement, I would suggest and propose the definition might need to change versus what's in Webster's. If we're truly talking about diversity of engagement, if we're talking about fluid engagement, the modalities of engagement, and engagement at certain contextual points. What does that look like and from whose eyes? And are we all clear of that? Whoever the we is in that conversation and all the voices. I would suggest and propose there are many in communities that was articulated so well earlier that are able to, yet might not be able to translate they're able to the systemic and structural things of able to. And when the points are connected, person to person, there's a natural catalytic event that takes place of that anticipation with aspiration, with purposeful intent. Because the engagement does take place. Because for families to be able to endure what they endure, both positive and negative, there's still a desire to. That's my unsolicited at 1:51 California time.
Great. Thank you so much, Bobby. Go ahead, Carrie Jo.
Yeah, no, I just want to add something on this point because I think that it's really important, engagement in education and trying to demystify, at least from our perspective, the regulatory process that can lead you to help contribute to a successful drug development program. And I think from our perspective, what we can take away is that programs like CEDARS, like the Accelerating Rare Disease Cures Program, education is an important and critical pillar to how we view, being able to move rare disease drug development forward. And how are we going to share that with people? Well, we've worked really, really hard to try and put together a website that has workshops on many of the topics that are shared challenges. So every rare disease, condition and program has its own unique challenges. It's one of the things we've talked about today, but there are shared complexities across these programs, right? And there's a lot to be gained from learning from others' experience and learning from sort of the common principles of drug development, because those you can apply. And so we've really worked hard to put together a website that has resources for multiple workshops on all the things we've heard about today, like the general considerations, the collection and use of real-world data, the development and use of biomarkers, novel endpoints, and statistical approaches for small populations, and how that can be challenging, and some things that, like you heard today from Dr. Kidwell, ways to overcome those. We've also heard that people can't find the information from us that they want, and so we've through like our learning and education to empower rare disease drug development program, heard feedback that people wanted the guidances. So now we have a guidance tab there, collated by topic, so people can actually find what we think on various topics more easily, as well as funding opportunities. How are you going to get started? How are you going to fund your natural history study? We have a number of grants and research programs available at the FDA, and those are all there collated in one space. So we're trying to set up a one-stop shop for some of the learnings on regulatory processes that we have together, so that people can find them.
Great, thank you so much,
Carrie Jo,
Carrie Jo.
once they've found them and once they've used them, because I have gone through that page and it's quite helpful. What happens with the 2.0 version, right? When you finish FDA 101, how do you engage on 201? So you've gone through the rules and regulations on how advisory committee works and what the conflict of interest rules are. And you have now got a proposal for disclosing conflict of interest and creating a non-voting role for an actual treating physician in a super rare disease. And so how do you engage the FDA on that suggestion after having read the material that's on there?
So the advisory committees are actually run out of a different office at the FDA, but they can't, you could engage, I think there's an FRN out right now with an email for how to engage on those. The Cedar ARC program is focused on like the scientific and regulatory considerations for developing a program. So,
Yeah.
and what goes into that. So the workshops on there are the topics that I had mentioned before that are real challenges for the community as they're trying to develop programs or contribute to developing programs.
I think we're talking past each other,
apologies.
two different things. Yeah.
Yeah, apologies. I'll have to, sorry to take moderator's prerogative, but I know we've only got 6 minutes left. We do have, I think, a question that I think would be a good way to sort of tee up final thoughts, final comments from the group. So I'd love to sort of just jump to Carrie Jo really quickly, maybe move to Colin, Jennifer, Karen, Syra, and then Bobby. So I'll go in that order, so you've got a heads up. But sort of a final question, how can we ensure patient engagement activities really are meaningful and impactful, that they're efficient for patients, sponsors, and regulators moving forward? So would love to hear just final reflections on that. Carrie Jo, I'll move to you just to see if you have anything else to say before moving to the others.
Yeah, sure. Just really briefly, I think I'll go back to something that I think Bobby brought up. This is one of the things that we've heard today is an underscoring of the tenet that, you know, everyone living with, affected by, or working on one of these 7,000 to 10,000 rare diseases, there's a community. This is a community. It's a shared community, and it is one that benefits greatly from dialogue. It takes all of us working together. Every one of us has a role to play in drug development. You cannot do this alone. You cannot do this in silos. It's too hard and it will take too long. It really takes patients and families who drive us with their lived experience, right? As well as the patient advocates, the researchers, the clinicians, the sponsors, the regulators, and everyone has a voice. I think you've heard how much that voice means to us, certainly at the FDA. We do take that into account when we review and design our programs, but also for the sponsors designing them and for the patients to have a voice in that role as well. It's really critical to have early collaboration in listening to the patient voice, because you have to be thoughtful when you're planning clinical trials. You do not have large populations. You may not have multiple phase studies. You don't have a chance to do over in terms of a clinical trial if it's not well planned and well thought out. You have to produce, you know, evidence that can be utilized to evaluate the effectiveness and safety of these therapies. And I think I'll just end with saying that meetings like one that happened today are so valuable for us, because we really do love to hear from diverse and multiple stakeholders, because our goal is to take this back and inform and enhance our outreach, our materials, help the community better understand how to contribute to and design rare disease drug development programs that are fit for the regulatory framework. Because in the end, we all have a shared and common goal, and that is to make more safe and effective treatments available to patients.
Great. Thanks so much, Carrie Jo. I think I said Colin next, so go ahead.
Yeah. Mine always, I sort of echo Bobby's comment in that I always look at this from a clinician's perspective. You know, back in clinical days, that's why I got into research, and it's really sitting down and almost with the golden rule, you know, or treating someone like it's a member of your own family. You know, making sure that you're respectful of their time and energy and opinions. You know, don't be paternalistic, you know, in where decisions. And so they're like, oftentimes I thought I had the right answer, you know, that we wouldn't do this, but needing to actually listen. And make sure that you're contributing back to the time and energy that they're giving you, I think is really important. And as we tread in the space, I work in a lot of patient communities, and it's one of the things I'm most sensitive to, because some of them are progressive diseases where every moment they're giving me is a moment they could be doing something else. And so making sure that I'm also, you know, being an active listener, you know, very strategic and thinking about how I'm going to use the information that they give me, because they're giving me their gift. And I want to make sure that I honor that.
Great. Thanks so much, Colin. And I know we're going to probably go a few minutes over. So I appreciate everyone being patient as we make sure to give everyone sort of chance for last comments. So Jennifer, go ahead.
I'm going to repeat myself and say partnerships, because it's so important to be engaged in the process. I was really pleased to see multiple presentations today, including Dr. Bracchio, show how this engagement process is going to be continuous over the development life cycle. And so that makes it so important that When advocates and caregivers and communities come together to contribute data, experience data, participate in trials, natural history studies, that we know we're going down the right road, that this information will really be informative to regulatory decision making, because otherwise we're wasting time and we're wasting money. And our community gets fatigued. They get fatigued from participating in surveys and natural history studies. We have 13 years of natural history data, and we just paid to keep this study going for 3 additional years because we think it's so important. So we need to make sure we have the right outcome measures and assessments there that will really inform development in the future.
Great. Thanks so much, Jennifer. Karen, go ahead.
I agree with everything that was said. I think really going into these conversations with the mindset of being in it for the long haul, drug sponsors, patient families, patient groups, and FDA really not just thinking about the problem at hand, but the long-term relationship that is being built. And I think that goes back to a lot of what Colin said about respect and really treating each other with the deserved respect, and trying to understand each other and the challenges and unique frameworks within each of us operating, and really thinking of as Bobby said, as people first, and really bringing it back to that conversation, to that long-term partnership. I think ultimately that is what helps break down a lot of the miscommunications, the misaligned expectations, and that ultimately helps make this a win-win situation for all of us.
Great. Thanks so much. Saira.
I agree with everything everyone has said as well. I guess I would end with a note of incremental, thoughtful, tangible, small changes are really helpful in the bigger picture as we take the long view. So let's make what changes we can incrementally and thoughtfully, so that every day it's a little easier and a little easier. Long views are good, but sometimes they feel like we're boiling the ocean and then we get stuck. So small incremental changes that are tangible for patients.
Great. Thank you so much, Saira. And Bobby, last remarks.
And my brief comment in air quotes, considering that I've lived with all my stuff and to where I'm at, and yeah, anyway, 52 years, is both the elephant in the room, cockroach in the room, and beagle, seagull, and eagle. As a child, there was an elephant in the room that I could see from many different things that were negative. And the roach, they flip back and forth. Yeah, cockroach. Ray killed them eventually, okay? And to get me to where I'm at now. And to have a beagle, as in the dog, seagull, the thing that fly and it's there in the water, and eagle's view. There's a significance to all of that. Let that sit and marinate with you for a little bit, like a good soup of beagle, seagull, and eagle. If each is able to marinate with that, going into the next meeting or discussion or space or place of about building together, that may help in the process.
Great. Thank you so much, Bobby. And with that, I really appreciate everyone's time, all your thoughtful comments and remarks and specific recommendations for really how to move forward in this space. And really just want to thank everyone who's participated today. I'll turn it over to Marianne Hamilton Lopez to really help close us out.
Right. Thank you, Victoria.
Thank you.
All right, I will try to be brief. I know we went a little over, but wow, we've covered a lot and have gotten such an amount of rich comments in the QA especially. So just to review some themes, we heard very clearly in our trial design discussion that for a trial to be effective, patients and caregivers must be meaningfully involved in the design process from an early stage, so that the other stakeholders involved can understand their day-to-day experiences living with a rare disease and can design a trial that works for them. During the next session on case studies, we heard about the value of and the challenges associated with developing and incorporating natural history studies. Also, the need for bidirectional communication with the FDA that can help patient groups better understand how their input has factored into overall scientific review, and opportunities for FDA to foster consistency across review divisions and centers. And just now in this last session, We heard about some great progress that has been made towards improving patient engagement with many different stakeholders, including both industry and FDA. Importantly, we also heard about priorities for continued improvement. The importance of bidirectional communication was emphasized again, as well as other ways in which expert input could be brought into the review process, and the value of all stakeholders being very clear and transparent with regard to how and when patients can best contribute, and how those contributions will be used across development and review. And I'd like to emphasize that this is not the end of the discussion on this topic or the last chance to share with us your feedback related to this meeting. We and our colleagues at FDA will be continuing work on these critical issues, and the public comment docket associated with this meeting remains open until February 12th of 2024. Again, the link to that docket can be found on the Duke Margolis event page for this workshop, and we hope to receive additional feedback from many of you there. Now, before we go, I do want to thank everyone who contributed to this excellent workshop. First of all, thank you to all the speakers and panelists who shared their expertise with us over the course of the workshop. I want to especially thank the patient advocates who shared their personal experiences with us today. You were not here to talk about something hypothetical, but instead you came to talk about how to improve drug development for your children, your loved ones, and yourselves. That's not always an easy thing to do on a public forum, and especially one with hundreds of participants. And so we greatly appreciate your involvement. Thank you to all of our colleagues on FDA's planning team for this workshop. And then also a big thank you to my team here at Duke Margolis. You saw many of them on the screen moderating today, but those behind the scenes, I want to thank Thomas Rhodes, Dure Kim, Mia Williams, Hannah Vitelio, and Luke DeRocher. Finally, just thank you to all of you in the audience. You were engaged, you were thoughtful, you had questions, and a dialogue that went in within just a chat. That's what makes these workshops so successful. And we really greatly appreciate your involvement. Thank you for joining this workshop, and have a great afternoon.